Bioinformatics, a specialized analytical wing of biology of life science. And nowadays, this is one of the subject trading in research, development, and job market. It is now so much in hype that even Google have Google Genomics. So we are here for this workshop to gain much more knowledge with respect to this particular field. So to start off this session with blessing from the divine energy, I request Manormai for the invocation.
विघ्नेश्वर निनगे प्रथम प्रणाम स्मरिसुवे निन्नय दिव्यनाम विघ्नेश्वर निनगे प्रथम प्रणाम विद्याधरणे विघ्न निवारक वंदिते निनगे जग संरक्षक विद्याधरणे विघ्न निवारक वंदिते निनगे जग संरक्षक निन्नय महिमेय निलियुगे प्रतीक्षण बेडुवे निन्नये कृपयनु अनुदीन विघ्नेश्वर निनगे प्रथम प्रणाम गंगाधर सुत चंद्र सुधारक मंगल गोणिसो श्री शुभकारक गंगाधर सुत चंद्र सुधारक मंगल घोड़िसो श्री शुभकारक भवसागरवनु दाटनु अनुदिन भवसागरवनु दाटनु अनुदिन सार्थक वेतव कारक नाम विघ्नेश्वर निनगे प्रथम प्रणाम स्मरिसुवे निन्नय दिव्यनाम विघ्नेश्वर निनगे प्रथम प्रणाम प्रथम प्रणाम प्रथम प्रणाम प्रथम प्रणाम.
To illuminate this session, I request all the dignitaries to light the lamp. Now I request Professor S.S. Malini to welcome the gathering.
A very good morning to all of you. A warm welcome for this auspicious occasion of inauguration of workshop on bioinformatics. First of all, I would like to welcome all the dignitaries on the dais and off the dais, all faculty members from Department of Genetics and Genomics, research scholars, students, teaching staff, non-teaching staff, all of you. It gives me immense pleasure to welcome our Honorable Vice Chancellor, Sir, Professor G. Hemant Kumar, to this auspicious occasion. He is a man of visionary, a man of simplicity, a man of vision, and he is a great human and great personality. So because of his constant support, we could able to conduct this workshop, and he has given financial support. And his support started when the department has started in the 2018, and in 2012, I hope I remember, he has provided some softwares also to do extensive work in genetics. And till now, he is showing his unconditional support to all of our activities. And it is not an exaggeration if I speak about him, because he has started Career Hub, that is a skill oriented and to get more opportunities for all our students in the campus to get a job in good companies and IT sectors. And that is his dream child, and it's working fantastic. And many students, they got jobs in a different sector. And he has given promotion to more than 400 teachers, which is pending from almost 9 years. And to this, his credit, lot of such changes he did in the campus, and always he's supporting all academic activities in the campus whenever we approach, and he's extending his support and encouragement to all of us. And with this few words, I request Dr. Chaitra to welcome him with a bouquet. Now, to grace this occasion, we have another very important guest personality with us, Mr. Raghu Rangaswamy. He is Vice President of Indian Operation, joined Schrodinger in year 2004 as a technical support scientist, and has been promoted to his current position since 2013. Raghu manages business development and administration of Schrodinger India office. He was a university topper, gold medalist, and received his master's in pharmacy from MGR Medical University and completed his MBA and PG diploma in Bioinformatics from Madurai Kamaraj University. Prior to joining Schrodinger, Raghu started his career as an assistant professor in PSG Medical College and worked as a bioinformatics scientist in Biocon, DSQ Biotech, and Tata Excel. Raghu is well known in bioinformatics and molecular modeling community in India, and he sits on the academic advisory board of many Indian universities. He is also a trustee and secretary to Friend of Champion India, an organization dedicated to mentally challenged children that shows his concern towards society. Not only in his profession, he is also extending his services to society. And with this, a few words, I request Dr. Somana to welcome him with a bouquet. Once again, it gives me immense pleasure to welcome another known person to all of you, Dr. Amritavalli Madam. She is motherly in the department. Anything, any support, she's always extending her hands. It's not exaggeration. If she has started her own company, by this time she would be like a Kiran Majumdar. Should have done, but we are very fortunate that she has not started a private company and went back to private sector. She is in the campus, and whenever we need anything, she is supporting us. With these few words, I welcome Professor Amritavalli Madam, convener of this workshop. Dr. Pratibha, to welcome her with the bouquet. And I always feel it's a pleasure for me to welcome my mentor, Professor L.P. Ramachandra sir, BSR Faculty Fellow from the Department of Genetics and Genomics. And department is his dream child. And with heart and soul, he developed the department. And that baby, tender baby, is transferred to me. And it is my duty to take care of that baby properly, well fed, and that baby should come up with a beautiful color. And I wanted to uphold the name of the department at national and international level. That much of great responsibility has handed over to me. Sir, I never disappoint you. Definitely, I tried my best to fulfill all your dreams. With this, I welcome Professor N.P. Ramachandra sir. I welcome Professor G. Subramanian sir. Always upper bundle of the department. Any problem, immediately sir will be there to help us. That's why he's a upper bundle. Anything, immediately, instant that he will be ready. Sir, I welcome and I am very thankful to you all your support. Thank you, sir. I welcome Professor Rangarajan, sir, a man behind the lady. Amrutavadi madam. He's a highly dedicated person, professor from mathematics, and he's gracing this occasion. Thank you very much for sir for gracing this occasion. I welcome Dr. Basappa, former DR from the university. I welcome you, sir, Dr. Basappa, sir. I welcome all the faculty members who are involved in this workshop. Dr. Tejas, I welcome Dr. Tejas with a bouquet. I welcome Dr. Somana. I welcome Dr. Kaushik. I welcome Manjana. I welcome D.S. Chandrasekhar from Computer Science Department. I welcome one and all for this great occasion. Hope we never disappoint you. Definitely you will be having feast of knowledge all these 7 days, and it will be a memorable event in your lifetime. And I will assure you that this will be a memorable event in your lifetime. Always you remember this workshop whenever you take up your career. With these viewers, I thank everyone. I welcome you once again. I welcome ICT members. Media persons are here. Photographer Bhaskar and Just Kannada person is here. All media person, everyone. Once again, I welcome all of you. Thank you.
Now I welcome a very enthusiastic chairperson, Professor S.S. Malini, for this occasion. Now I request all the dignitaries to light the lamp. Now I request Dr. Amrita Valli to give the details about this workshop. Good morning to one and all. Honorable Vice Chancellor, Professor G. Hemant Kumar sir. Today's guest of honor, Sri Raghu Ramanathan. Chairperson, Divorce in Genetics and Genomics, Professor S.S. Malini. My dear colleagues from other departments, workshop participants, and media friends. Before I go into the details of the workshop schedule, it is my privilege to say
few sentences regarding the legacy of the department. In 2005, MSc in Genetics course was started in US in Zoology under the stewardship of Professor H.K. Rangnath, former Vice Chancellor, Bangalore University, former NAC Director, which was supported by the University of Mysore. Later in 2007, Professor N.V. Ramchandra led this course as the coordinator, and currently now Rangnath student is N.V. Ramchandra, Ramchandra student is Professor Malini, currently heading the department. This is the legacy that is inherited like a genome. So with this effort and then the support of the Vice Chancellors, the Department of Studies in Genetics and Genomics was established as an independent department in 2015. 12 batches have graduated the MSc course, with nearly 80% of the alumni are very well placed in premier institutions in India and abroad. In 2019, the department also started a unique prestigious course called as Postgraduate Diploma in Personalized Genomic Medicine, funded by DBT. So with great gratitude and regards to Honorable Vice Chancellor, I would like to thank him for continuously extending his support for our department and also today's workshop with financial sanctions. Sir, on behalf of the Genetics and Genomics family, we are extremely grateful to you, sir. Regarding our expertise to conduct this bioinformatics workshop, University of Mysore was one of the first universities in India to start postgraduate diploma in bioinformatics in the year 2001. Since 2001, University of Mysore has conducted nearly 10 national workshops, and nearly 400 MSc, MTech and BE and diploma students have done internship and project in our university in the field of bioinformatics. And nearly 30 PhD scholars from our department have critically used the bioinformatics tools in their research and published papers in prestigious journals. The faculty of the department are very well versed with bioinformatic tools and techniques. This is our background. So coming to the workshop details, the workshop is organized against the request of research scholars and teachers from all over South India. So we have restricted the participant number to 30, looking with the facilities in the department and also the COVID norms. And also for one more reason, individually you will be monitored and mentored during the training period. So the workshop is scheduled in a systematic manner to train you from fundamentals of bioinformatics, introduction to databases, Python programming, protein 3D modeling, and also drug designing from experienced expert faculties from the department and also from the industry. This is the outline of the workshop, 7 days workshop that you will be going through. So we have a very huge, beautiful campus with pleasant ambience and greenery. We hope all of you will have a comfortable and healthy stay in Mysore. Thank you.
Thank you, ma'am. Now I request Mr. Raghu Ramaswamy for the inauguration speech.
Okay. No problem. Thank you, sir. So good morning, everybody. Good morning to the Vice Chancellor and to the team for organizing this wonderful workshop. And I heard many people have come from different parts of the country. It shows like, okay, things are changing from a pandemic to endemic, right? And it's really good, like you people are organizing this workshop because this kind of things are not taught in the regular syllabus, right? That's what I'm going to talk. Like the way, like, okay, how the things are changing. You are conducting a wonderful bioinformatics department and the course for the last 7 years. Today, the way is how the things have changed from gene to drug. And what are the developments happened in the last 2 or 3 decades? What are the hurdles? And how the computational simulations are making it in the right and easy way. It's not working. Standard. Okay. So let me give a very small intro about Schrödinger. We are about 700 people employees. Like we are already in India about 70 people, but we are going to expand to 250 people in just next 1 year. So the kind of expansion like what we are bringing shows like, okay, how you might not imagine like, okay, the kind of developments like, okay, more than 200, 300% we are going to increase in the development and the bioinformaticians we are going to hire, computational biologists, computational chemists we are going to hire and everything. So we are not only into the drug discovery, we are also into materials, artificial intelligence, machine learning and all the physics-based modeling, everything. We are a 30-year-old organization which is started by DISHA. Now it is well funded by Bill Gates, Google Ventures and everybody. So we are not only a software developer. That was a scenario about a few years back. Now we do our own internal drug discovery and also many collaborative drug discovery programs. So we have so many deals like something like with BMS, we signed 2.7 billion deal, GR Lab and so many other things. We have already like in a short time, we have about 5 FDA approved drug molecules. Very, very short time. You all know like the drug discovery takes a lot of time in taking a better video. So we all know the drug discovery takes 15 years and takes about $4 to $5 billion. But today, like the scenario is changing, and if you see the kind of collaborations we have in the last 6 months, like with BMS 2.7 billion, with Viva Biotech, with Thermo Fisher, with AstraZeneca, with Bayer, like with Takeda, again with AstraZeneca, Q and Co, Morphic Therapeutics, Rexia Therapeutics, like MD Anderson, GR Lab, Google, and all those things. So what does this mean? Like why all these big pharma companies, big companies are coming in collaborating with a software company to do their drug discovery? Today, the world is changing much, much faster. You see, like probably the people like my age or probably like about overall like about 30, 35 years might be using like this was in 2000, like, okay, having this was a famous one. Then came a kind of thing, BlackBerry. Today, like, okay, we are all talking about Apple, Samsung kind of thing. The way, like, if you see like from a wired telephones, today, like, okay, this is all changed to like wireless. And if you see an iPhone or things, all apps like, okay, this displays so many things, right? So change is part of the way, change is part of the life, right? And you are all quickly adopting whatever the changes are coming into it. Same like, okay, if you're sitting just as an ambassador, like you're out. And at the same time, like in Mahindra XUV, like who booked like 75,000 cars in 1 hour. So if there is a change coming in, people are willing to change and you have a business. That's the way like today technology is changing. And if you look into the technological change in the life sciences, if you see like right from 1998 to 2003, it was the Human Genome Project which came in. That gave a kind of a big push for everything in the life sciences and the drug discovery, everything. If you look into the drug discovery pre-2000, it was more of a serendipity, right? Every pharma company, like, okay, was synthesizing different molecules. They were approaching different things, going for a combinatory synthesizer where they can synthesize even 1 million compound, but like without knowing what is the target, where it is attaching and whatever it is. So what happens? You have to naturally spend like 15, 20 years to discover a drug molecule. You have to spend $4 to $25 billion to discover a drug molecule. So good to the genomics, the Human Genome Project, which came, that gave the boom as a bioinformatics thing. Then it's not only the genomics, then the proteomics came, like, because you should know, like, okay, which protein, how they are communicating, how they are signaling, everything, right? Then came to, okay, you know the sequence, but like, okay, what is the structure of the protein? What is that like contributing that, right? So then the proteomics came, then the structural bioinformatics, the protein structures, everything came. So from a scenario in the last 20 years, if you see, okay, you have identified your gene, like, okay, you patented your gene, everything, like, okay, it helps to identify your function. Now, from there, like, okay, today, like, if you're looking into in the pharma industry, the way the major users of identifying these genomic technologies and everything, how the protein structure. So today, like, if you see the protein structural development, like, phenomenal. Today, again, with the AI machine learning coming in, again, this number of, if you have a sequence, like, okay, you can easily find out the structure of a thing, right? But what is that? Like, okay, have you identified a protein structure? Whether your job is over, you can discover a drug molecule faster. No. There are again challenges, like the molecule should be more potent, very good selectivity to its target. It should be soluble, it should be bioavailable, it should have a good half-life, it should have a good permeability, it should not have any CYP inhibition, hub blockade, it should be easy to synthesize, it should not be toxic. So many properties a molecule has to satisfy. When you have to satisfy so many things, you cannot go and use the same serendipity way of your discovering the things. You have to think of out of the box. What are the way? Like, okay, now, like in a drug discovery, you want to modify one, like, okay, when you want to modify any one of the property, you are modifying a functional group. When you are modifying a functional group, what happens? Because of that, like, any one of the things will change. And not only that, our body is 70% made up of water, so everybody knows. And the water in an outside environment and in a protein environment is totally different. Because the body, the proteins are like, you know, like in a simple way, like, okay, hydrophilic and hydrophobic amino acids. So if it is located in a hydrophilic area, then like, okay, it will have a much bigger affinity. If it is located in a hydrophobic area, then it is going to repel. So any molecule coming and binding to your target protein has to displace these water molecules. So once you understand the thermodynamics of these water molecules also. So without knowing all these things, how to solve all these things, just by like, okay, going for a serendipity way. So is there a rational way, right? That's why like what I'm coming to talk. So discovering a drug molecule is like, okay, it should be precision, right? If you're opening a lock, even if your key is a little bit bent, you're not going to open, right? So even if one property is not matching, you are not going to discover an ideal drug molecule. FDA is not going to give a thing. Because if anything happens tomorrow, like, okay, you have to pay billions of dollars and like, okay, FDA is blamed so many things, right? How can you like overcome everything? So this is a question asked, like, okay, to Arjuna, like, okay, what are you seeing? Everybody told tree, branch, this, that, everything. But he told, I'm looking at the neck of the bird, right? So when you're looking at the discovering a drug molecule, how much like, okay, you look into concentrate on like what is happening between a drug and a protein in the binding pocket, what are the contributing factors, what is the water thermodynamics, how can I design very effectively? And again, like in the last 20 years, the technological developments, like, okay, you want to cut a tree, you want to use an axe, like it is something like a simple computer, or like you want to go for a big server or a cloud computing, because how you are going to speed up your discovery, that matters a lot. Like when COVID scenario came, everybody is looking at, is there a drug molecule, right? Nobody knows, like it takes 15 years, right? So how can you speed up the discovery process and how it is going to help in the today's scenario? So if you want to discover something fast, okay, then it is going to be inaccurate. But if you want to discover something you want very accurate, then you have to spend time. So there are so many technologies, but today something like, can you combine everything? And today we are all talking about artificial intelligence, machine learning, deep learning, all those things. Combining these things, can you speed it up and bring out a drug molecule? That's what is going to be my talk today. So this was something like we did it about like something like 9-10 years back. We screened about 1 million molecules, and using all the computational things, we were able to come down to narrow down to a molecule with less than 10 nanomolar, and we sold for $1.2 billion using just a simple docking technology, right? But using other technologies and other things. But today, can you screen only 1 million molecules? Is there a way to screen billions of molecules? Because from like 1 million, can you take a best? Or from 1 billion, can you take a best? You can take a best from the billion because your data set is big. Now, when you have like AI, machine learning and everything as what I was talking, so even if you have 1 billion molecules, You take a billion molecules data set, take limited amount of molecules, do a simple docking and see using a machine learning approach, why this molecule is binding effectively or why this molecule is failing. What is the interaction between this molecule and the protein? Or what is the structure activity relationship, which we all call as QSAR, right? What is the structure and what is the relationship between its activity? What is its relationship to how the molecule is getting absorbed, distributed, metabolized, excreted, or toxic? Why this molecule is toxic? Based upon a structure, based upon these machine learning approaches, today it is revolutionizing the drug discovery, right? So you take a set of molecules like, okay, do a docking, like, okay, why this molecule is binding to the target protein and generate a model. So when you have generated a model with 3 or 4 runs with a good R square, then you can screen billions of molecules within few days. So even if you have a simple 25 computer processor computer, you can do a screening of 1.2 billion compounds within about like 20 days. If you have like something like 1,000 CPUs, you can screen billion molecules in just 2 days. This is unimaginable. Like few years back, like if you want to screen a billion molecule, like okay, you will be taking months or years. We screen 200 billion compounds in just 2 weeks, which is equivalent to 2,000 years of wet lab. What I'm telling you is today with the advancement of the technologies, the time reduction is so drastic and you can identify the potential molecule very shortly. Now you have identified your molecule. Okay, this is your potential molecule you have identified. But what is its binding affinity? Because the whole lot of things, if you look into it, how a drug molecule binds into the protein, it's all like, okay, how the molecule enters, how it displaces the water molecules, how it binds, what is the delta G? The delta G is contributed by so many parameters. Can a simulation do this? So once you have identified a molecule and it is binding very effectively, what you're looking into it, you're just modifying some functional groups, right? You're just modifying a functional group. So like this, like, okay, you modify from this like to this pentyl group, you're just adding an oxygen, or like you're just changing a CF3 group or whatever. So by these changes, what is the change in the binding affinity? What are the other parameters is this changing? Because the drug discovery, as I told you, it is a multitude of problems you have to address. So how we are able to predict? You see, this is the experimental, this is the predicted one. 5.3, 5.6, 6.5, 6.1. 6.4, 6.6, 6.5, 6.3, 6.1, 6.5. Such accuracy today computational technologies are able to deliver. If we can able to predict this within about 4 hours with the GPU computing today, a chemist problem is solved. Chemist is changing these functional groups without knowing whether it is going to change my binding affinity, whether it is going to change my, increase my solubility, whether it is going to increase my selectivity. Without knowing any of the information, he is blindly changing with his own intuition, right? But like today, if you can predict with the AI, machine learning kind of things, or with the free energy physics-based methods, and so accurately within 4 hours, that changes the game of drug discovery. And again, with the understanding the water thermodynamics in the binding pocket. So here is some of the case studies. We identified one single potential molecule, understanding the water thermodynamics, just 4 molecules were synthesized. Understanding the free energy perturbation, how the molecule is binding to its protein structure, like just 10 molecules. So within about like 14, 15 molecules, your drug molecule is discovered. It's not only for us. You look into it. This is from AstraZeneca. This was presented in a Biosim conference. Look into on different targets, how many molecules they synthesized. Just 10, 6, 48, 26, 82, 38. What is the R square? In only 1 or 2 it is 1.2 kilocalorie. Rest everything is 1 kilocalorie. Look into it like, okay, are they adopting these technologies today? AstraZeneca's invest like 15 to 20 million on the software technologies on Schrodinger. The way it is changing because they have seen the result by just synthesizing 6 or 10 molecules, they are able to identify a molecule for their target. Bayer, like, okay, you see like 60 ligands for 3 different targets, and overnight they are predicted. The way things are changing. And look into this is Johnson & Johnson. This molecule, I already have a molecule in phase 3. These are the experimental one. These are the predicted ones. You see like minus 7.7, minus 7.8, 7.1, 7.6, 9.3, 9.8. Why do you take 6 months to synthesize a molecule? Here within 4 hours, I can give you the result as close to what is your wet lab, which also has the same error rate. So this is Galapagos. Just 15 molecules, 16 molecules. Here prospective 100 compounds calculated, 10 recommended. Just 3 synthesis, all calculated perfectly. Now, what I'm talking, even if you have a billion molecule, okay, this is the database of billion molecule, filter based upon diversity, cost, and so many whatever the parameters you want, reduce to 100 million compounds. Use the AI, artificial intelligence, or machine learning, which is for like how the molecule is going to, how the absorption, distribution, metabolism, excretion, everything is there. You have created so many models based upon the known molecules, or using the QSAR, QSPR methodologies, reduce from 100 to 10 million molecules, do a quick docking, reduce to 1 million molecules, then come to the water score, understanding the water thermodynamics, glide, prime mmGDs, which calculates little bit more accurate binding affinities, reduce to 100,000 ideas, then use the free energy perturbation, which helps you to identify when this molecule is binding, how the water thermodynamics are changing in the binding pocket, how the protein structure is changing, how the protein ligand interactions are happening. This what takes just 4 hours, and just from 100,000 ideas to just 25 ideas. And with the 25 ideas, you sit and see like, okay, what are the best molecules I can synthesize? Just 5 molecules. Every PhD student working in a chemistry department at least minimum synthesize how many molecules? At least 200 molecules you synthesize, right? To get a PhD. How many molecules? Why can't you implement a technology like this? Just synthesize 5 molecules, not 5 molecules, even 25 molecules. You approach rationally the technology, whatever is there. Bring a molecule and IP, right? For the university. Go and sell it, whatever it is the thing. That's the way the world is changing today. That's what I'm telling. Like, okay, you look into why there's so many big companies are coming and collaborating, right? This is the way, like, okay, the development of candidate, whatever is happening. So today, we are giving you like, okay, now I know like madam is arranging this event, the workshop. Good, because like these are not taught, these technologies, whatever I'm talking, these are all not in your syllabus at all. Your syllabus changes after 20 years. But the way like, okay, the way technology changing, like a mobile phone changing, like if the CEO of Nokia was telling at the thing when they sold to Microsoft, we don't know what mistake we did. Only thing is we didn't innovate, right? When the change is happening from a touch phone to the other technologies, we didn't change so fast. We were sitting with the same technologies, right? Like this is what is happening. This is not in your syllabus. Good, like okay, you all took pain traveling from so many distances to come and learn what is the technology. It's not just conducting a 1 week workshop is going to give you a complete knowledge. It should be part of your syllabus. It should be implemented in your every project. What is the new technology coming in? Okay, is there an AI machine learning coming in? How can I implement in my PhD project or in an MSc thesis? So that helps you not only just go for a 1 impact publication, you can go for 4 or 5 impact publications because you are using the rational approaches in your whatever the research problems. I'm not talking about only on the drug discovery. We do the same thing in the materials. We do the same thing in the formulation. Today, like Boeing is collaborating with us, like okay, to find out like okay, what is the low weight flight I can design so that the consumption is less. Today, Samsung is collaborating with us to bring in the new technology TV, how to bring in the blue light and everything. Formulation companies are innovating, collaborating with us. How can I design an effective formulation when the patent is getting over? How can there is no formulation aspects designed in that, given in that. Like how can I bring an effective formulation? Same in the biologics, protein engineering. The technologies are changing. We are able to predict so accurately. So today, like we want to bring in a center of excellence in selected universities. If somebody is interested to collaborate and innovate with us, so where everything, whatever I discussed, you will be getting it. You can access anywhere from the institute. This is a few of the universities which are all having the molecular modeling, chemistry, biology, bioinformatics, pharmaceutical research. We are going to select only 10 universities. We already have about 3 universities in the list. And you can introduce in the master's, PhD projects to produce high impact and IP, bringing everything. We also provide every 6 months, we come here and do a workshop for about a week, 1 to 2 weeks kind of a workshop every 6 months. Not only that, complete 24/7 online support. We work with every project, every PhD project, and all that. So we have about 400 scientists who are working in drug discovery collaborations. They will also collaborate and work with you, where if you are interested in the center of excellence in the molecular simulations. So no doubt, now we are in an era where we are really able to reap the benefits of the computational modeling, use the vast amount of molecular modeling tools. Today, computational simulations are playing a very crucial role in the drug discovery, materials discovery, formulation, we name anything in the bioinformatics, structural biology, everything. It's time to implement in your day-to-day research for a quick, accurate results, which helps to bring in many publications, patents, discover your own IP, everything. And it's time to consider the molecular simulations seriously in your research. So this is the paradigm shift. Gone are the days of like, okay, synthesizing millions of compounds using a combinatorial synthesizer. It has a limit of at the max to synthesize a million compound. Today, using the computational, you can screen billion. The days are not far. You can screen even trillion because of the cloud computing coming in. Use screen using the target identification, target validation, everything. Bring to it. So when you have to address a multitude of problems, you need multiple tools. You want to go on like roam around like with the same way of this and lose the time and effort, or like be smart like Ganesha, because ultimately you want to get the drug molecule, whatever it is, quickly like with a limited effort, right? So decision is yours. Good, like you are taking effort to organize these kind of workshops, but just not 1 week is not enough. Use it, practice, and use it in your day-to-day research, day-to-day thing. India is a country like, yes, we have brought in rocket technology. We are so great, space technology. We are called as a pharmacy of the world. But are we having a capability to discover even a single molecule? We buy the raw material from China, just make it as a tablet, sell it to the Western world. But tomorrow, if China stops, like, okay, no, I'm not going to send the raw material, our farmer thing is gone, right? So we should like change from a service-oriented country to a research-oriented technology company. We should innovate, we should bring in. That's what like the universities has to change. It's good, like, okay, the Vice Chancellor is also hearing this. He is the one who established this in the Center of Excellence earlier in the same building. You had it. See, there is one thing like, okay, the students here, like, okay, with the great efforts, people like them, they bring these facilities. But today, in fact, like when he was doing a PhD, he might have struggled to even to get a one publication, to get one paper. Today, the data is, enough amount of data is available, enough amount of facilities available, enough amount of opportunities are available, enough amount of distractions are also available. Are you focused? Are you taking the right opportunity, like, okay, for your project and doing it, right technology and doing it? It's good, like they're organizing. I'm really happy for them. And yes, you're taking a first step, but try to bring in like all these technologies into your day-to-day research. Thanks a lot for giving me an opportunity.
Thank you so much, sir, for systematically explaining the rationales of drug design. Now I request our Honorable Vice Chancellor, Professor J. Hemant Kumar, for his presidential address.
Hello, namaskar. Very good morning to you all. So I'm very happy to be here for this inauguration function of the workshop on bioinformatic tools and techniques. Today, just the chief guest, Sri Raghuran Swami, who just delivered his keynote address, and Professor S.S. Malini, who is organizing secretary of this workshop, and Dr. Amrutavalli, convener of this workshop, and Professor Rangarajan, Professor Ramchandra, so Professor Subramanian, and our deputy registrar, and all of the students and faculties of the genetics and genomics department, and other faculties of the other departments, and all the researchers, students who have come from different places, and media friends. So this workshop is for 1 week. So being the technology is being grown, where the computers and softwares which we have, as Raghu rightly pointed out. So when we were students, just to have the research papers in not very long ago, it is 92, 93 or 93, 94, something like that. So our computational power was in Institute of Science sometimes previously. So though we had computers here, so to get the reference material, we used to go to Institute of Science. So morning till evening in the library, I used to search. So I used to get 20 papers. So just to finalize my research, what we call problem. So then we used to spend 300, 400 rupees. On those days, it was not a what we call small amount. So then we used to do the things. At that time, all my computations used to take, as Raghu mentioned, we had only P3 computers. Technology was that much. In P3 computers, I used to get the result, one program, one result, one set of results. For 2 days, it used to run. So, this artificial intelligence and other things we studied when we were students in 86, 88. So, though it was there, it was an algorithmic form. So, we used to develop the algorithms, we used to put the things such that it was all on theory, but we could not implement it. The implementation was not possible because we didn't have powerful technology or powerful what we call the processing units. Processing units we started with what we call the least processor 8086. That was the computer with extended technology, PCXT. So with that our work started. And before that we had TDC 316 in our lab. So it was third generation computer, university. I think we have kept some components here as exhibition. So then we had mainframe computers where the CPU itself was what we call half of this room size. It was there with us, where defense DRDO gave us. So we used it. So then later on in 1995 onwards, the technology for processing units. So thanks to that, electronic engineers who processed it, who what we call invent. So that type of fast processing units came. So though we had the software was not executable because we didn't have what we call the power for processing units. So we have P5 and we have Pentium and many what we call the functions are there. So even a small mainframe computer, it's a big mainframe computer, is replaced by a small unit. So that's how after that we could able to realize this artificial intelligence and machine learning because it needs a lot of computation. This computation can be realized through what we call a computing unit which was working very fast. So this artificial intelligence and machine learning on those days we used to teach. People were not liking. I used to give what we call some of the elective courses when I was a child. Only 5, 6 students used to take. So now if I say AI, 100 members will come because it is executable now. Is it? So you had this in your research also. So a lot of things like analysis of the sequence of DNA, RNA, proteins, and all those things taking a lot of time. So you have got the bioinformatics using computer programs and a variety of applications and determining the genes and protein functions in establishing what we call evolutionary relationship and predicting 3-dimensional proteins and other things. So relevant to these advances, recent advances in field in bioinformatics, it is possible to use the power of the machine learning and data science. Now machine learning everyone uses. So once upon a time, it was invented 1970s, 1960s. It is a part of statistics. So this learning started from statistics, mathematics. So it was able to realize because of the electronic research. Nobody appreciates that. So finally, we had this all the programs ready, search methodology and all the algorithms was ready with the computer science. Now any subject can use it. As Raghu mentioned, it is in bioinformatics, it is in material science, it is in docking. So many what we call things we can use it. So what we call this once upon a time any computer is a very large computer. No, it is not so. So we need not hire any, not buy any computer. We have got cloud computing. So cloud computing technology. So we need to have a good software related to your field. So that is what the Challenger is doing now. Challenger is taking all your problems and putting onto the computer and producing what we call the software. We are all using it and what we call developing the new technology. And whatever we are doing in, as you told, in 200 days, 2 weeks, we are doing it in few hours. Is it right? So that's what the technology is. The technology we need to use it. So for that, the drastic now, the current this thing is using AI and ML. There's a drug discovery and development, and we need to have produced the drug repurposing and also thereby improving the pharmaceutical productivity and clinical trials. So today we need few hours to prepare what we call this new generation, new generations for the computation purpose. The students and scientists will integrate in a multidisciplinary knowledge is required, and there is what we call use of modern bioinformatic resources, resource powered with advanced operating systems and also database and networking technology. Because once you change the hardware, related operating systems should change, related what we call the software related should also change. So I wish that this 7 days workshop conducted by Schrodinger, that it be a useful one. The thing is, university is ready to invest. Thing is, what is the output? And personally, I know this docking software will be purchased. When first time Raghu came, I told that no, it is not possible. Then many of our scholars and teachers came, no sir, it will be useful and we are using it. I think on those days we gave 35-40 lakhs for those softwares. It was used for 1 year or 2 years license CA. And now what we call, there is no demand for that. Nobody came to what we call extend that. But some papers we have published. That should not be. Say that we are ready to have center of excellence, no doubt. But all our teachers and researchers should come forward. Sir, we buy this in 1 year, we are going to give 100 papers. I am going to buy it. So it is not single department commitment. Many departments should work together. So we are ready for this pharmaceutical research, drug discovery research, where some leading interdisciplinary team should be developed in the department. I think Professor Ramchandra knows, Subramanian knows how hard we worked for this biodiversity project. To execute this project in this building 100 crores. So all many departments work together. So I wish that in future days, a team for this drug discovery and also this pharmaceutical research should come forward. If they are ready to say that we are going to make a group, we commit to for 100 papers in 1 year. So definitely we are going to establish your center of excellence, not with that amount, 50% discount we have to give. Okay. With this, I used to say that interdisciplinary is now this national education policy is also what we call increasing interdisciplinary research. So we should not be so far away. So once upon a time, the technology was not available. Though it was there in different countries, it was not available. Now it is readily available. Only thing is we have to pay money. And once you pay money for the software, every moment is important. Because I am buying the software for licensing 1 year. In 1 year, we have to use it. Next year, next year license again. Again, we have to pay. So that type of things, if they come up, and in future we will plan for that, and it is a new year. So let this workshop be successful in giving a good dimension and direction to all the researchers here. So in future, we will have some more workshops such that all our researchers will be able to use these type of softwares on hand so that what we call it will be more useful to them. Thank you very much. All the best to you.
Thank you so much, sir. Now I request our beloved Vice Chancellor, Professor Hemant Kumar, to felicitate our Chief of Guests.
Sir, they should encourage 40% ಕೇಳಿ sir. Sir, 40% ಕೇಳಿ. They should encourage the institution more than.
Sir, I'm giving you 40%.
Now I request our chairperson, Professor S.S. Malini, to felicitate our vice chancellor. I request Dr. Pratibha to hand over the fruit ball to our chairperson. Now I request Dr. Pratibha to deliver the vote of thanks.
Good morning all. It gives me an immense pleasure to deliver the vote of thanks for this event to all dignitaries assembled here. I would like to thank our beloved Honorable Vice Chancellor for his financial support and Professor Shivappa Sir Registrar for his constant support and guidance. I do thank our Chief Guest Raghu Rangaswamy Sir, Vice President, Scotinger Bangalore, despite of his busy schedule, for accepting our invitation to grace this occasion. So I would like to thank beloved and dynamic chairperson of our department, Professor S.S. Malini ma'am. Thank you, ma'am. I thank Professor N.V. Ramachandra sir, who's our chairperson and is now a PSR faculty fellow of UGC. And Professor Rangarajan sir, who is the chairperson of mathematics department. And I would like to thank Professor Basappa sir, who is the faculty from organic chemistry department. I thank ICD and IOE for the facilities they provided. And last but not the least, all our faculties, research scholars, and non-teaching staff, and all our students. Thank you all.
Thank you.
dose of caffeine, right? I hope so you're all active now. So I would like to spend some more time, like, to address any of the questions, whatever you have. It can be any nonsense questions also, don't worry, because I'm going to be here for about 15 minutes or half an hour. Okay, I'm not going to judge you or anything, but like I know the younger generation has lots of doubts, questions. What should I learn? What should I do? Or like, okay, I explained some of the things, but you might have not understood some of the things. Even if such is the case, like, okay, you feel free to ask me whatever it is. So any questions? Yeah. You have the mic there?
Yeah.
Sir, what are the recent advancement in AI?
In AI. See, AI, as your vice chancellor told, it was an old technology, no doubt about it. It was like those days it was called as neural networks. Today, but those days what happened is, if you want to find out a property, if you want to build a model, the number of data what you have is was less. Okay. Today, if you look into the recent developments in the world, say why Microsoft, like which was selling like their programs for what are the number of dollars, or today they are willing to give it free. Today you take Facebook or WhatsApp or any of these things, technologies. They're willing, okay, use my things. Okay. Why even Google Maps, which is worth multi-billion dollar, right? WhatsApp, the company bought for $18 billion. Facebook bought for $18 billion a few years back. So these are all given free to you. But at the same time, you all understand, like everybody is collecting the data, right? And all this data are used to somewhere, like again, back target to you, right? So when it came to drug discovery or any technology, see today, if you look into AI, like okay, look into the disease models, like okay, what are your clinical data, they can predict like okay, whether you're going to get into a disease or whatever. So the insurance companies will say like okay, I'm not you have a chance of like getting this. Right? Even tomorrow, genomics is going to be like that. Today, whatever the genomics today, it started as bioinformatics, like the few sequences, whatever we had to identify what is the function or whatever. Today, like that famous actress like Angelina thing, like she removed the breast, right? Based upon what? Just by doing a genomic sequencing, like and identifying a particular gene which can cause a cancer. And I assume like tomorrow, like this data is going to become very, very important. Okay, tomorrow, like okay, if you want to do the things are going to change, like okay, you do your genome sequence, and insurance company may tell, okay, give me your sequence data, let me tell you, like okay, what are the chances of you getting into cancer or hypertension or diabetes? Okay, at this age you will reach this, so I will give you insurance only up to this age. So when it comes to AI, all this data, how they can get the data and like use the data in everything. When it comes to drug discovery, so we were approaching, in fact, the drug discovery started with the kind of AI. If you see like in 90s or 2000, it was all, the famous things was like 2D QSAR, 3D QSAR, all those things. It was analyzing a particular property of a molecule. See, every structure, activity as an activity. And there is a relationship between every structure and its activity. Like, okay, if you're changing from a chlorine to bromine or from a methyl to ethyl, that might be changing its solubility, binding affinity, or blood-brain barrier permeability. If you're increasing your hydrophobic functional groups, like, okay, it is going to increase its hydrophobicity, so it can reduce its solubility, it can cross its blood-brain barrier. So there is always a relationship, right? But like if you have only 10 molecules or 100 molecules, how can you predict? It is not possible, right? At the same time, like when you have more number of molecules, like let us imagine you have thousands of molecules or millions of molecules with the molecule, with its activity, and like, okay, you bring in a correlation, like based upon like, okay, this molecule, I'll split it into pieces, okay, number of, how many carbons are there, nitrogens are there, like what is the molecular weight, what is the log P, log S, different things, and I can correlate with its activity. With this molecule, if having a structure like this activity would be like this. Or its log P, log S would be like this. It's going to cross a blood-brain barrier, or it is going to have a toxicity. When more number of molecules came in, so today we are able to build a better model. Okay, so that's the same thing happening in the drug discovery. So if you take drug discovery, earlier it started with all these things with the quantitative structure activity relationship to predict the activity. And then like, okay, the 2D, the two-dimensional base, then three-dimensional base came. Then slowly the technologies like the rational drug design came, because the development of the bioinformatics, identifying the genome, identifying the structure. Okay, now we have a structure, okay, can we go and design based upon the structure? Right? Earlier, like you don't know what is the structure. You are just relying only on the data, right? So that was like kind of a information-based technology. Now when the structures came, okay, here is a structure, well, let us go and approach a problem rationally. Okay, what is the protein structure? What is the water thermodynamics in the binding pocket? Okay, which water molecule is thermodynamically stronger? Which is thermodynamically weaker? Okay, so when a molecule is binding first into the target protein, so it looks into like it has to displace the water molecules, right? So which water molecules are replaceable? Which are not replaceable? That's what we do it in a rational drug discovery. So whatever is displaceable, okay, this water molecule is displaceable because probably it is located in a hydrophobic area. The water doesn't want like the water like which are located in a hydrophobic area, that is not going to have a good binding affinity because water is hydrophilic, right? So that is easily displaceable. So when you can displace that and keep a hydrophobic functional group, right, that is going to increase the binding affinity. If it is like located in a hydrophilic area, the water molecule is not possible to displace, right? So hydrophilic attraction is there. So you cannot even extend the functional group. If you extend the functional group, like what happens, like the molecule cannot sit effectively. But at the same time, like this same information, if you are looking into kinases, where the structural information is so close, all the kinases has a similar structure. So analyzing the water thermodynamics helps a lot. So this, like, okay, all the kinases are similar, but in this kinase, there is one water molecule which is thermodynamically weaker. So that I can displace this water molecule with a favorable functional group, so I can design effectively. So if you look into the drug discovery, there is one is rational approach where you do the docking, analyze the molecules which can fit in perfectly, come to the water thermodynamics, understand the water interactions, do the free energy perturbation. The free energy perturbation is nothing but like, okay, in a docking technology, we are keeping the protein structure fixed. Okay, so this is fixed. Like I am taking a billions of compounds. I'm trying to see like, okay, how each and every molecule fits, how their interaction is, what is the contribution of hydrophilic, hydrophobic, addition, donor, acceptor, van der Waals, electrostatic, every interaction between the molecule and the protein, and what is affinity it is having, so that which molecule is best having a higher score. It is like, okay, you want when you go for your higher entrance exam, for IIT or whatever, right? Okay, how well you scored in your plus two, like how good you got in the physics, chemistry, mathematics, biology, or whatever. If you got a good marks and everything, then okay, you are the top candidate. Same way, like how these interactions are. But in docking, we keep the protein structure fixed. But in reality, the protein is flexible. As I told you, the water thermodynamics plays. So that's where like the dynamics comes. You have to consider the dynamics aspect. That's where the free energy perturbation comes. Like, okay, when a molecule is binding, what is the amount of water molecules are going to go out? Which water molecule is stable? And in a protein being flexible, how this molecule is going to interact? Is there a change happening in the binding pocket? Is there some residue changes happening? And how this protein and the ligand are accommodating? What is their binding effect? That is called free energy perturbation. Earlier, it used to take at least 6 months to do one free energy perturbation technology. It was a old technology, no doubt. But today, tweaking of that, and also like with the GPU computing, the GPU came like with the gaming, right? Because if you want to do a gaming, you need a faster speed and everything, right? Because the movements are very fast. So that's the same thing came into the molecular dynamics, into the drug discovery. So one of the aspect is like, okay, today, as I explained, you can predict the binding affinity of a molecule within plus or minus 0.5 to 0.7 kilocalorie. But identifying only the potency is not your drug discovery is over. There are toxicity, there is ADME, absorption, distribution, metabolism, excretion, CYP inhibition, hERG blockade, everything. But the thing is, you have got so many molecules today, researched over so many years, which this is the molecule, this is the toxicity. These are the groups like, okay, contributing for the toxicity, or like these molecules having this kind of a functional groups can cross a blood-brain barrier. So you have so many molecules, so many data is available, right? So with these data, create a model, generate a model. Okay, when you are generating a model using this machine learning, artificial intelligence, so you have generated the models. So these models helps to address the non-rational based approaches. So when you have a structure, you can predict the binding affinity, you can predict the solubility, you can predict the selectivity. These are based upon the structure. But whether a molecule is going to be toxic, whether what is it's going to be, how it's going to be absorbed, how it's going to be distributed, we don't have any structural data, right? So we cannot approach it on rational data. So there the data is available. Take the data, model it, and predict it. So today artificial intelligence is playing a very major role in every aspect. If you look into the retail, it's playing a very crucial role. If you look into the banking sector, it's doing wonderful things. So same thing is happening in the drug discovery also. And today, if you see like a few years back, oil companies were their valuation is so high. Today, the company which holds the data is so valuable. If you look into the Facebook, what is the valuation of it? If you look into the WhatsApp, what is the valuation? Because what they have the data. And with AI coming in, that's what is picking up. Yeah, next question.
Hello. We have several complex diseases. We shortlisted certain genes by screening, SNP, exome sequencing, whole genome sequencing, or microbiota technology. I have nearly about 50 or different, at least 10 different diseases. For example, Parkinson diseases. So the family member, the kid, sons and daughters, do I get a Parkinson because my father and mother had this accident? So we have identified certain, we shortlisted them also. We have those. Now, even we identified not to the extent that they are ligand or they are the secretory protein, they are transcription factors and so on. We have classified them also. Our PhD has gone to that level. Now, I have this kind of the gene sequences of the some of them do have a protein crystal structure, some do not have it also for our understanding. So this is a good molecule for the drug to be developed in these cells. So how best your company can help the university, and many people have this kind of thing. How best we can have understanding and we can help us. Please.
Okay.
To
Now
do
one
something.
yes, okay. So one is like now let's tell you are telling like you have genes, okay? So you don't know whether it has a structure or not. Even if you have a structure, fine. But even if you don't have a structure, if your sequence, protein sequence, has a sequence similarity to any other protein sequence which has a structure 70%, even not 70%, even if you have 40%, it's enough. Okay, we can model the protein structure. Today, with the AI coming in, there is a program called AlphaFold. Even if you don't have a structure, also like okay, it is predicting based upon the AI-based thing structure it is predicting. But again, you have to bring in the computational thing because what happens is if you take into a structure, there is alpha sheet, beta sheet, alpha helix, beta sheet, and the loops. So the alpha helix and beta sheet you can model it really easy. The loops are the more flexible ones. That you can model based upon the modeling tools. And with the structural biology catching up so much, probably these things also can be screened, predicted so fast. So once you have these things, then you have to come into if you want to discover a drug molecule, you have to come into the rational way of approach. Where if you have a structure, yes, okay, you can screen like okay. So again, databases of molecules, there are enormous, like synthetically feasible compounds, or even like you don't have to go and synthesize also. There are like companies which says like okay, these are the molecules I can supply to you. Okay, you take this database of molecules, you just see like whether it docks or whatever it is, whether it fits perfectly to your target, screen it, take the molecule, okay, do your initial wet lab thing because you just order it, you get the molecule within a week or 10 days, screen it, you're using a wet lab, okay, it is having a good binding affinity as per close to your thing. Okay, now look into the molecule, change whatever the functional groups, whatever you want. Then again do the same like free energy perturbation, whatever the technologies are identified, and then you can identify quickly. But today is, if you look into the life sciences, drug discovery or whatever, it is not your one single man work. You have to collaborate with a chemist, you have to collaborate with biochemistry, you have to collaborate with SAP today, like if you look into the pharma industry, it's not one person doing it. There is multiple departments, right? And if you look into any good publication coming in Nature or whatever, so many, like there are 25, 30 names are there, right? Yes, like, okay, you have the data of the genomics, get to the bioinformatician, okay, can I get a structure for this? Okay, go to the molecular modeler, can you fine tune this structure? He has predicted it, like, can I fine tune it? Go to a chemist, like or a computer molecular modeling person, which molecule among this, like, okay, I have the, for this gene, like I have the structure for this. Then, like, as a chemist or a molecular modeler, okay, the chemist, like, okay, now is it synthetically feasible? Now, like, okay, what functional group I can change? Then go back to the modeler again, like, okay, he has changed this group, how well it is binding. What are the other parameters, like, okay, this ADME parameters, toxicity, and other parameters, how well it is matching. Today, we have developed a tool, like, which is called Live Design, where it is an interaction between all of you. It is an interaction between the wet lab, it's an interaction between all the chemists, biologists, everybody. So whatever a modeler, whatever you think from a wet lab point of it, okay, you put this, you put that structure. Okay, they can model it. The modeler can suggest everything. Even the chemist can interact and say, okay, I want to change this functional group. He can immediately click of a button. He can say, like, okay, how the molecule is docking, what is its binding affinity, what are its other ADME, other parameters, because based upon the models, whatever you have generated and kept it, he can predict everything, and in a short, within few minutes, you can take a decision whether I can go ahead and synthesize. That's the way the technology, computational day is changing.
So adding to the, in addition to Sir.
Okay, again your collaboration. So that's where, like, I'm coming to the center of excellence, where we are interacting with. So in any case, also we interact with every customer. We give our best support. Not only on training the software. So if you are having a problem, like, okay, I've done this. Can you suggest me, like, okay, what is the best way? Have I done it right? Can you suggest me, like, what is the other things we can do? So we have about 9 scientists in Bangalore, and we have about 70 people in Hyderabad, and we have a Europe team. So we can all, like, there to interact and support.
Yes. So in addition, so artificial intelligence and if we presently the scenario, if we look into it, so now we all know, so the remdesivir was initially found for Ebola virus. Now it is very effective for our COVID-19, so which targets basically the RNA dependent RNA polymerase. So my thinking is that, so this type of drug repurposing, which is now like a quick decisive matter, where if something really needed, we use the drug database and do some repurposing and getting it into market, because we already know about the drug toxicity.
Yes.
So how can you comment? How is your opinion about advantages and then disadvantages? Because I will tell you, curcumin, some other days we used to say, oh, it's a wonderful drug. But now recently, as a chemist, many chemists shows that this binding is very off target. It is binding and it is having a lot of other interactions. So in this regard, positively or negatively, what is your take home message to the future generations?
So if you want to bring something effectively quickly, okay, drug repurposing is the best, because everything is tested. Okay. But the drawback, like why nobody gets into the drug repurposing, is where is your patent? Right? It's already patented by somebody, right? So the patent is over. Let us imagine aspirin. Okay. When aspirin was discovered, like, okay, it was for a different, but later on, like, okay, today every cardiologist prescribes an aspirin. But like its patent is over. But like by that time, like, okay, if they have predicted, okay, aspirin is a good blood thinner, probably like they marketed like so much. So the only thing when it comes to drug discovery is, can I discover something quickly, and like, okay, can I sell it? Like if you look into a Western world, so okay, I have discovered this, whatever price I want, I can go and sell it for next 15 years. The moment like any market molecule goes out of the patent, the price drops 95%. And like hundreds of people are competing with that. With the 95%, they're still marketing and they're making so much of money. So it all comes into like, okay, if there is already something existing, okay, it can cure. Say today ivermectin, they say like it is effective COVID thing, right? And today At some point it came in, yes, it is effective. Some point they told like, no, it is not. Again, yesterday I read an article like ivermectin is very effective against COVID. Same like curcumin, like no doubt, like okay, it is an effective anti-inflammatory, antiviral, everything. But if you see like the problem of curcumin is like its absorption, right? So, but like okay, whether it goes and targets exactly the same thing may not be, but probably it may go on like act on some anti-inflammatory markers. It can reduce the anti-inflammatory effect. Because of that, like your other things can subside, right? So you don't know like exactly you have to hit only that. You can go around somewhere like that's why like in the drug discovery pathway also we are looking into the pathway like where you can hit, like okay, where you can hit to the lowest most. But on the other way, like you can also go into like if there is a hypertension, like okay, you can target like with the different calcium channel blockers or ACE inhibitors or different kind of thing. At the same time, like if it is due to inflammatory, like okay, you are even reducing the inflammation can also reduce it. Right? There is multitude of problems for any disease. It's not like only one thing. So you can address it in a different way also. See, if you take Ayurveda, what they say, Vata, Pitta, Kapha. That's it. Three things. Can you like if any imbalance happens here, like okay, it happens there. They address on that, like they address to the root cause of that. In allopathy, they address to the target. Okay, it's only to maintain. You take if you have hypertension, you take anti-hypertensive for lifelong. You will diabetic, you take for lifelong because you are going and inhibiting at that point of time, and then like okay, it's you have to take it rest of the life. That's the way the Western medicine works. Yes.
Dr. Sir,
Yeah.
like during pandemic time, every company were, like they were manufacturing, they were trying to manufacture drugs or vaccination. So during that time, Schrodinger's company, what they contributed during the COVID time?
Okay, that's a good question. So okay, what we did is we did a collaboration with Takeda, Novartis, Uzi, and one more company. So we were doing a philanthropic based drug discovery. So we screened about billions of compounds. We identified some potential compounds, and it is the Novartis is testing because they have the wet lab facilities for the, because when you want to test any antiviral drug, you need to have a good antiviral BS3 lab. So they were having those labs. They were testing it. It's undergoing the clinical trials. So anyway, like what happens is like in the drug discovery, you can speed up the lead identification optimization. That's what like you can speed it up. But you cannot speed up your animal studies. You cannot speed up your clinical trials. They take their own time. Okay, they might take like okay, probably 3 years, 5 years, and then like okay, by the time like okay that comes, probably like in the antivirus, the problem is by the time the molecule comes, the virus changes. So you saw like okay from COVID to Omicron to Delta, like okay, so many different things, right? There is structural changes happen. That's problem with the antivirus. So we did a philanthropic thing, even like we decided if a molecule is identified, it is going to be free.
Excuse me. Working on extracting biomolecule active molecule
From plants.
Yeah, from either plant source or the synthetic compounds. Also, first we'll be working and we have a lot of biological data. So we want to know like next step, in silico analysis, what all the in silico analysis can be done for the molecule and how much it would cost us.
Okay, that's a good question. Again, if you look into it, most of the drug molecules which came out in the last so many years, for a kind information, is plant-based. If you go and see the literature. Second, how you can use the in silico approach, because I'm having a plant molecule, whether it is going to have this activity or not. How do I identify? There is a company called Atrimet in Bangalore. So they are our customer. When they came to us a few years back, he is an Ayurvedic doctor, Dr. Damle. He came to us like, okay, I'm an Ayurvedic company. I want to use your in silico tools. I was a bit taken back, like, okay, because we have, you name any CRO, any R&D, pharma R&D, they are our customers. First time a plant-based company is coming to me, they want to use the in silico tools. I told, okay, so what are they going to do? So he told, I have a big database of all the plant-based molecules. Okay. So I wanted to see like, okay, when I want to screen, because there is something like, I want to do a correlation. So in the real COVID drug, like there is a molecule called Vivus, they are marketing it now. I would say it's really effective. In fact, like I don't want it to because to be a marketing person for that, but we really use it. I personally used it. My family entirely used it. It was really fantastic drug molecule. Okay, they have now backed up with the clinical trials. But I'll tell you like what they do. They have a huge database of all these plant-based molecules. So what they do is they use the in silico tools, the docking and the induced docking technologies to screen these plant-based molecules. Now, when they screen these plant-based molecules, okay, you don't know like which molecules it is. The plant-based molecules comes from different sources, right? So we don't know like, okay, this molecule is going to have what activity or whatever. So when they screen it, so which molecule is binding effectively? So this molecule is binding. Okay, now this molecule is coming from which source, which plant? Okay, and what is the earlier literature available? All the 5,000-year-old literature available, what it says, what are the uses these our forefathers are like our rishis or whatever, what they've written for this molecule, for this plant, what diseases it can cure. Okay, so then he identified those plants and he extracted from those things and he created a capsule. He went and did for the wet lab studies, human trials, everything. Now it is marketed as a effective COVID drug. So this is one success. I can show you many, I can share success stories.
In addition,
But this is the way you can do.
Yeah. Thank you. So in addition, as a researcher, I would like to get your query that. So if you have a biological property and your molecule structure is known, so you better go online where many tools are available, where human targets could be predicted for that structure, and mode of action can also be detected. So we can give you so many even what is the range.
So if you have a molecule already, like okay, we have another
Yeah.
tool like which is called QuickProp. What it does is it goes and it predicts all the properties as well as it also compares with the already existing drug molecules, how close this is to it.
Yeah.
So that also can give you some idea. Okay, this molecule can have this kind of an activity. It is not based upon this to see like in genomics we do phylogenetic analysis, right? Okay, these genes are similar, like okay, what similar, how close they are, kind of thing. Same like in the chemoinformatics we have, like okay, this is a drug molecule. Okay, you can go and compare against the thing, like okay, which two are very closely similar.
Yes. Even
Okay.
we can filter that. Homo sapiens, we can filter Plasmodium falciparum or COVID. So even China have some good software online where for COVID they openly open source the drug prediction. Okay, so where you can put in your structure and see which is the target. Okay. So this and all will you go into PubMed and see what you want and see the in silico approaches and the original articles which are really reported to do something. Okay. Yeah.
Which one? Screening? No, in silico analysis, like if you have the software, like it is, it doesn't cost you anything.
Yeah.
You have the software here, like okay, it doesn't cost you anything. No, you can do it.
Yeah, you can do it yourself or you can write.
You can ask like Dr. Basappa, like okay, he can just take the molecule, you have a simple molecule, he can just do a docking and give you. That's it.
Yeah.
No,
That's why I told you like you have to collaborate with others.
Yeah. You can write to us, so we will give you my email ID. So we will help you. Yeah.
That's what like these people want to get it also. So you have to get into that. That's what is the next. Like yes, genomic era was there, is there, no doubt, and it is going to take a different dimension going further. Genomics, to my knowledge, bioinformatics and genomics is going to evolve in a different way. In the future of identifying the disease, these things, everything is going to come up. But at the same time, like okay, as what you are asking, if you have identified something, how you can take it, in silico comes into it. You can do the in silico. Yes. So in silico will give you supportive data. Okay, so that's what they are also doing. So when they want to go and approve, put it for approvals, so all these approval agencies will ask, okay, what is the proof? Like, okay, this is an Ayurvedic drug, your plant-based drug is coming. What is the proof this molecule is binding to your disease sample? You show me the proof, right? So where is the proof you have? Like, okay, you have wet lab. You can say like, I have my wet lab data, but where it is binding, you tell me. So when you want to show the proof, like, okay, then the in silico has to come in. Okay, this is the way the molecule is binding. These are the interactions it is doing. This is the binding affinity this molecule is having. We can predict and show like, okay, this is what it is. And this is what is matching with my wet lab ITC data or whatever. That's the proof like where in silico comes in.
In fact, the journals are asking mode of action.
True.
Or the structure.
But like if you want to go to Western, the Western looks into it, the proof data. Our forefathers didn't bother about it. They gave like all the human clinical trials, everything, and tested with so many people, and they taxed like bus. That's it. Take it.
Now, in silico mode of action is, madam, which is very important. We don't want to go with the serendipity. Okay. So some of our article you can read. So AI based target prediction tool. We call it as some kind of like very basic one called my mentor Markprint 2D. So which has been used for a longer time. So such a programs have been used by Schrodinger and so many commercial softwares also available. So simply what I used to tell you, you drop an email, you will let me know.
Sure, sir. Thank you so much.
Yes.
Good afternoon, sir. I just want to know what skills do we need to be a good bioinformatician? The skills we need.
Okay, if you are looking into bioinformatician or whatever, one, understanding what are the technologies coming in. You should also be able to do a good programming because some of the things you need a good scripting knowledge and all those things. Okay, and whatever the tools, technologies coming in, you should be an expert into it. Okay, you should be able to use the latest technological things coming in, understand the basics of it. Why are you doing it? What are all the algorithms? Okay, if you're doing a sequence analysis, what is a BLAST algorithm does? What is a Blossom matrix does? P52 matrix does? What is the fundamental? How they are identifying a sequence identity, similarity, or BLAST, FASTA, or whatever? What are the basic things? What they are doing? How they are searching and finding out? That's the kind of things. Like you should understand the basics of it, and it's not just like a click button thing, right? Okay. Why, like, okay, where you have to use FASTA, where you have to use a BLAST, okay, where you have to use a PAM matrix, or what you have to use the other matrix algorithms. So you should know that like exactly, and the scripting knowledges. If you want to come to molecular modeling, you should understand like, okay, the chemistry, the structural information of it, and also like about the force fields, like, okay, whether you have prepared your protein perfectly, whether you have prepared your ligand perfectly, because what you're doing the simulations are like in the computer has to be similar the way like how it is in the biological environment, right? So whether you have made sure like, okay, if you're taken a molecule, whether the protonation state, then the molecule is binding, whether this is in the correct protonation state, whether the charges assigned are perfect, like whether the software assigned is correct or not. Okay, then like if it is not assigned, like, okay, there is something like we have force field predictor, like, okay, you can assign the force fields, or you can do a quantum mechanical calculation and assign the things. So you have to make sure like before the starting point, everything, the chemistry is perfect. Whether you have prepared the proteins, because what you get from a protein crystallography is just without a crystal structure, without any adding the hydrogens or histidine tautomeric states and that things. You have to prepare, make sure like, okay, you have assigned the histidines, histidine tautomeric state, the hydrogen atoms are added, and the amide flips. All those mistakes are corrected, and it is prepared perfectly before you go and do a document. Okay, and how you do the other technologies. If you are preparing an AI model, like, okay, what is the kind of data, what from which researchers, is this from the same lab, the experimental condition is same, like all those things you have to give a priority in generate, and again which statistical algorithm you are using. Okay, today like the technologies like we have AutoQSF, which predicts, which can even take like, okay, which the statistical algorithm is good, which can predict all those things, but like the basic knowledge is must. You should, like, when you enter into anything, you should know in and out of it, the basics of it. That's what is fundamental. Not don't just do a button clicking kind of thing.
Yeah, it is true that again your specialization is important. What do you want to be? Okay, you can run you can see or you can get exposed to a good software or anything. But what you want to be is also very important. Being like understanding neurobiology or cancer biology or system biology. So based on that, you can choose up some protocols or some software.
So being all youngsters here, let me tell you like within 5 minutes what I'm basically a pharmacist. Okay. When I pharmacy graduate, I worked as in the medical college. So what I used to always think, so I'm from biotechnology. So when I took my biotechnology, like okay, went and asked my principal, how is the biotechnology going to be? He told it is now, as of now, it is in a starting stage. Next 5 years, it can boom. Okay, what is coming in the next 5 years? I want to be into that. Okay, I settled. Now I was working in a medical college as an assistant professor, fine, in my own native everything. But I used to still go and read lot of literatures, lot of journals, so many things, because they are the one which is going to give you an idea of what is the next technology going to come up, right? So if you want to be employable, you should be saleable. Okay, you should get into something like, okay, what is going to come up in next 5 years or 10 years? What is the technology going to come up? So when I was reading in the library, I saw like bioinformatics. Okay, I'm talking about in 1997. So bioinformatics, okay, where is it? Where they're offering? Those days are like the internet was just booming up. There were like nobody is offering bioinformatics. So again, like luckily I was doing my MBA in Madurai campus. I went and asked like, do you offer bioinformatics course? Yes, only 2 universities are offering bioinformatics course. One is Madurai and one was Pune University. I went and asked like, okay, what is the procedure? Okay, this is the thing you should know, like, okay, I'm from biotechnology background, but at the same time, you should know programming and everything. I went and learned programming for 3 months, wrote the exam, everything. But my idea was like, okay, what is going to be next thing? Okay, the human genome sequencing is going on. At the time, only 2 universities were offering it. So I went, studied, it was a 1-year course. Before I completed my course, I got a job in Biocon. Even I didn't complete. So now again in Biocon, why I was sitting, okay, we have identified the gene, like okay, genomics, human genome, everything. We have identified what gene, what function, everything. What is the pharma industry is going to do with this genomic information? Because at that time it was just like genome, function, everything. Bioinformatics, that's what it is. Okay, now like in next 1 or 2 years, they will identify for all these genes, what is its function, like okay, what is its relationship, what its protein it is going to translate or whatever. This information is gone. What is the next one? Okay, now the structural genomics is going to come. Okay, and what are they going to do after the structural genomics? Okay, it's going to be drug discovery. What is this? Is there anybody offering? Nobody. Even today, nobody offers on computational drug design or anything on any course on in a master's level or anything. So what I did, like, okay, there was a software available in my own industry. I was working in DSQ Biotech. From evening 5 o'clock till night 2 o'clock, I sit and go and read all the tutorials. I took the computational chemistry videos. I read everything. I don't know what is docking. I don't know what is dynamics, what is pharmacophore, nothing. But read, like, okay, use the internet, like, okay, learn whatever it is. Go and do your projects. I went and gave a presentation in IIC. They told like, why you allowed this guy to come and present this in a student section? You should have presented him in the lecture, like, okay, where all the professors were talking in the morning. That's a be like, okay, one, like, learn what is coming up in the next 5 years, next 10 years, what is going into the gray area, right? Then, like, how can you be an expert? Whether somebody is teaching or can you teach yourself? Today, like, you can teach yourself with the amount of technological, with the YouTube or without, there are so many online courses on everything is available. Okay, it's not like, okay, your university has not changed your syllabus. Learn what is the knowledge you get. What is a project you can do? How can you showcase yourself? Like, okay, I am an expert in this. You design your own website. Today, like, I have a student who was studying in a school and +1. She came to me. I want to do drug discovery. In +1, she is coming. Okay, I told, okay, we will teach. Like, okay, the person is coming to us like an interest. Like anybody come who wants to do your research. You don't have a software, don't worry, come to us. We will help you. We will guide you. If you're doing a PhD, there are so many people who come who are doing PhD, having some problem, come to us. We help. There is nothing, like nothing to help. Okay, nothing. We don't charge anything. So I told like, okay, come learn. You know, she learned being a +1 and she did her +2. Like I saw, like I went to her website. She told her I developed the website. I went to her website. I saw, like I was wondering, like, okay, what kind of a knowledge she has. She has worked on so many things. She has done so many courses on Coursera and everything. And she got offers from so many years universities. She's studying now in years university. So be proactive. Learn what is coming up next. Pick up like how can you learn? Be an expert. That's where like you are scalable.
So I have a basic question, sir. So you said like even though there was no structure, the AI can predict the ligands of the target. So now like conventionally when they're doing homology modeling, you used to check the Z-score and the function and all those stuff. So how does it correlate? Say, when it doesn't?
Okay, what we are doing is a very rational approach. In the modeling and everything, we are approaching rational. What the AI does is like, okay, it is predicting based upon known with the knowledge of the other things, whatever it is. But if you still have, if you have a structure with about 70, 60, even above 50% of sequence identity, go for the molecular model. You can take that also, come back here and fine tune it. Well, that is just purely based upon the data, with the known structures data. No doubt about it, it is also doing a good job, but not as close to what is the reality here. Okay, but like when your percentage identity is so less, let's imagine you have only 20% of sequence identity. This will not do. Less than 30% will not do. Because we've got 70% of the data we don't know. Because all what we are doing is with a known structure, we are transferring the XYZ coordinates. There it transfers based upon the knowledge base. So you take that from there and fine tune it. So you have to look into it like where you can use what technology and everything. Okay, I'll take this last question and
Good morning, sir. I'm Manohar from JSS. I have a question. I think it's a very basic question. If it's a molecule and if it's a gene, we know the physical qualities and chemical properties of it, and we never know all the biological interactions that possibly it could do. If you can say in terms of percentage, on what percentage can we be very sure about bioinformatics when we use a chemical assessment?
Okay, this is still a gray area to us. Okay, if I have a molecule and where it can go and bind to a target. Okay, but as of now, if you ask me, like, okay, one way to do it is like, okay, you take whatever the drug, whatever the PDB databases are there, you can go and do a docking to all those structures, whatever you have. But if you see like today, like with the thanks to the bioinformatics, like okay, identify what are the potential targets and what are the structures available, but still like the structures for many targets are not available. But like with the AlphaFold AI coming in, like probably it may throw out so many. For if you have a gene, like okay, you can predict what is the structure going to be, and like if you want, like probably the days are not far away. Probably in few years, like we will be having structures for all the entire human genome, like kind of things. So if you have a structure, then probably and technology will come, like okay, where you go and do a cross-talking across all the proteins, to which protein it is binding, predict it, like okay. Not days are not so far, but as of now, it's in a starting stage.
Sure, sir.
Thank you.
Thank you so much, sir. Time is the constraint, or else we would have continued this session. Thank you so much, sir. Now I request Professor Subramania sir to introduce our keynote speaker, Professor N.D. Ramchandra.
ಒಂದು ನಿಮಿಷ ಇದಾಗ್ತದೆ ಅವ್ರದ್ದು ಇಂಟ್ರೊಡ್ಯೂಸ್ ಆದ್ಮೇಲೆ
Good afternoon. It is a great pleasure for me to introduce Professor N.D. Ramchandra. Professor India, who is going to deliver a lecture on genomic medicines today. He is an award-winning dedicated teacher and a professional academician, both in the contemporary genetics as well as human genetics and genomics. I am closely associated with Professor NBR, you believe it or not, since 1980. the day he joined MSc Zoology in the North campus 42 years ago. I'm sure he remains as a role model to the younger generation. It is a paramount interest to know how he has reached outstanding academic excellence by his hard work and dedication to duties. He is an intellectual and spiritual teacher mentor with the purest intention of seeing his students spread their wings and get to the skies. After his master's degree in zoology and doctoral degree in 1988, he worked as an ICMR and CSIR research associate for a couple of years, and later as postdoctoral student from 1990 to 92 at McMaster University, Canada. He also served in the USA and also in Austria for a couple of years. He began his teaching career in the Department of Studies in Zoology in 1992. I'm just a little bit elaborating it because the younger generation who joined the department, they don't know his biodata. So please take, I will take another 5 minutes. He completed long span of service as popular teacher. From 2007 to 2015, he served as a professor and coordinator of the Department of Genetics in the University of Mysore. And from 2015 to 20, he was the professor and chairman of the Department of Genetics and Genomics in the university campus. At present, he is a UGC DSR fellow, which is a prestigious award. One thing I want to say that he is actually instrumental in establishing this wonderful department of the campus, campus, that is Genetics and Genomics Department. Yeah, he has guided almost 28 PhD students, and I was just talking to him yesterday, out of which 15 are in the field of genetics and genomics, medical genetics. So, total 275 publications in referred journals by tapping a huge funds from DBT, DST, CSIR, UGC, ICMR, and whatnot. His H-index is 28, which is a noteworthy achievement. So, regarding recognition and awards, there's a big list. I know there is a positive plan, as Dr. Pratibha, Chaitra has mentioned, but I have to mention a few of them so that the younger generation should know it. He is a Sir C.V. Raman Ames Scientist Award in 2007. He's actually, Professor G.K. Manna Memorial Award in 2015. Rajaraman Award again in 2015. And actually, University of Mysore Golden Jubilee Foundation Award in 2016. Elected as Fellow of the Indian Academy of Sciences, FASC in 2017. And what now? And I just want to remember that what exactly he has served as a local secretary of 103, Indian Science Congress. Because of his hard work, his dedication and involvement, I think the Science Congress actually is one of the successful events, and many Nobel laureates still actually correspond with us. And he actually, picked up a few Nobel laureates during Science Congress. Sir, it's really a pleasure for me. I love so many things. Please don't feel otherwise. So, ladies and gentlemen, please join me in welcoming Professor Ramchandra to deliver his lecture.
Namaskara. Thank you, sir, for elaborate introductions. I thank my department persons inviting me to give talk, to share my experiences or somewhat what I have done. Two things before I start I have to take. Many times in the last 30 years, whenever I have to give talk, it will be posted. I will avoid to give talk in inauguration day. Most of the time, inauguration extent, my talk will be in a lunch time. It so happened in 2016 in IISc, my talk was started at 2 o'clock. The lunch time was 1:30. Okay, so otherwise it will be in the evening. at 7 o'clock or 7:30. So that was a challenge for me. Another thing that, as Raghu and others said, even our Vice Chancellor Hemant said, in 90s when we started our career as a faculty here, I told several of my colleagues that 92 I joined to introduce isolation of DNA in our department. I was not allowed. Simply say that it's not in our syllabus, you need not waste time. It took 2 years for me to convince my biotechnology department. First time I made my students to biotechnology student to isolate DNA, simple DNA. Then finally, 96, we introduced in our zoology department and all other department, just the isolation of DNA. That was the 30 years back. Not even biochemistry department, I'm not exaggerating. You can ask your senior colleagues, botany or biotechnology or microbiology was not there at that time. It was not there. And now we talk about the genetics or genomics at a high level, and we in our department store thousands of whole genome sequences, exome sequences. Every day, our colleagues and students work on them. This is the 30 years of growth in our department. Yeah, let me briefly discuss about what I'm going to do now. This is my outline of a talk. Is only very briefly I'm going to explain them about genomics and genomic variations, data analysis, and what level of the medicine we are getting it, and how genomic medicine is going to affect, and what is the future course of genomic medicine. I believe some of you might have seen this slide. This is my favorite slide. Always I put this slide. Can you read this? What is the answer? I believe all of you know genetics starting from 1865 by proposing the Mendel's law through rediscovery of Mendelism 1900 till date, the same laws are applicable, workable, and without that, any biological science cannot be taught. The understanding the Mendelian principles. And that's why I try to put forth all of you that genetics is the foundation of all life science courses. I know some of you can make these traits, the characters. All these characters are coded by the genes. We inherit genes from in the advent of technology, the scientists ask the questions, can you rectify this? Can you treat them? Can you manage them? Can you prevent them? That has started. Okay. In the multifactorial, when you look into the category of diseases, look at the other side here. Genetic susceptibility component is very less, whereas environmental factors are more to cause AIDS. This year, the genetic component, the CFTR G is responsible, transmembrane protein is responsible more than 90%, and the environment is about 5%. We do have a category of diseases that's a multifactorial. 50% is the environment, 50% is genetic factor, the hypertension, coronary heart diseases, and many diseases. Friends, only 50% of the genetics is involved causing any diseases, 50% is environment. Even though susceptible genes are there, depends on the environment, those susceptible genes or defective genes are enhanced and activated, become very severe. So as I already told you, single gene factors is responsible for cystic fibrosis, anti-stentoria, sickle cell anemia, as we are already aware. Multifactorial, environmental factor, the lifestyle and the genetic factors involved to cause a disease, and many factors are being involved. This is the more challenging for us to understand now. So with the simple techniques, with the simple understanding of the genetics, is what it was not possible to treat, manage these complex diseases. Now, with the advent of technology, genomics or personalized genomics is more helpful to even change or cure, to some extent cure these complex diseases. That's why the genomics is booming now in all over the world. So now, what is genomics? Most of you are aware, it is the study of the genome with certain techniques, analytical methods, and asking right kind of scientific questions. And genomics is the medium for the message for the 21st century. As you are aware, in our schools and colleges, the Human Genome Project started in 1990, completed in 2003, and even working draft of the sequences was published 2001, but it's completed 2003, and it amounts to an haploid genome over 3.2 billion base pairs. And it finally ends up with around 25,000 genes. We do have protein coding genes in our system. Even the public funded genome project as well as private genome project joined together to establish the complete analysis of the human genome, and even they identified what is the size of the chromosomal DNA and genes present on each chromosome. I want you to concentrate some of the wherever I mark that these chromosomes are arranged based on the cytological technique, G-banding or tree-banding methods. The smallest is the last, the largest is in the beginning of this. 1 to 22, they numbered and X and Y. If you look at the 17th, is smaller than the okay, one of my slide is missing the. But the first chromosome is having about 2,700 MB and about 3,000 genes present. if you look at the 17th and the 17th chromosome and the 18th chromosome is bigger than the 17th chromosome, but however, the gene number is small. That indicates that if the chromosome size is bigger, need not have more genes. And also, I hope you know that the 21st chromosome is a culprit chromosome for to cause Down syndrome, having around 303 genes. And the X chromosome, where all the females have 2 copies of the X chromosome, have about 1,100 genes. And Y chromosome, very few genes. Most of the genes have been lost during the primate evolution and hominid evolution. They have about 230 genes and the Y chromosome. If you analyze the complete genome, only 1.5% of the genome is coding, about 25% of the genome is intron with the intron sequences, and about 20% is the line sequences. These are the long interspersed nucleotide elements, and as well as many other repetitive sequences or the dead transposons are there. This is the ultimate the genome what we have now. After the Human Genome Project completed, many challenging projects have started, and because the one genome sequence is not sufficient, then they started understanding different ethnic groups, and many variety of genome follow efforts have been started with the individual genome project that Watson, James Watson sequence that we completed, many other individual sequences we completed. Craig Venter sequence inside the 2007 the individual genome. Like that, then they started 275 ethnic groups of HapMap genome sequence they completed. Then they proposed 1,000 genome project. Then they proposed 1 lakh genome project, 1 lakh Asian genome project, 1 lakh the UK genome project, even 20,000 Indian genome project also proposed, and they have been almost completing now. And they also comes with a personalized genomics, individual exome genome sequencing, million copies of exome and genome sequences available in the database now. Now, what is genomic variations? As I already told you, any trait or the gene sequences are changed and they become abnormal, and that leads to broadly a variation. A change in the genome sequence is a variation between 2 individuals or among us. The changes can be recorded. So what are the causes for genomic variations? Mainly the mutation. What are mutations? Many of you might say now sudden change, because it has been taught in PUC college or high school. The sudden change. The mutations are the changes in the heritable changes of the DNA. Okay, and when exactly mutation can inherit to the generation next generation, they are in the sexual reproductions, meiosis during meiosis, and as well as fertilization, because in fertilization 2 haploid genome meet and then the zygote forms and the whole organism will be developed. So mutations are very, very rare. What is the frequency of mutation? Spontaneous mutation? 1 in 1 million to 10 million. Only 1 in out of 10 million nucleotide, only 1 million will be changes will be there. They are cornerstones of this. If there is no mutations in the early life, and you would not have this diversity of organisms also. So mutations are there, they are fixed, they are there, different individuals, different groups, different life forms, plants, animals, and microbes have been, including humans, have been evolved. So the mutations are the raw material for the genomic variations. We have studied in the schools and colleges and the textbooks the chromosomal aberrations. They are deletions, duplications, inversions, and translocations. Now, with the genomic data, 4 more classification of the variations have been recorded. The single gene nucleotide, they are called single nucleotide polymorphism, there are SNP, only one nucleotide is substituted or changed, are deleted or added, and there's substitution that are SNPs. Then few nucleotides are added and deleted, they are called microsatellites, and about 10 to 100 nucleotides are added, they are called indents. There are insertions and deletions in the genome, and the 1 kb to 5 MB, the sequences are deleted in the genome or added in genome, and such category of genomic variations are called copy number variations. These are they are not there in the text of new terminology. One has to understand more about this. We know all these things, but we have to classify them, understand more about the to understand the abnormalities and diseases. For example, transversions we know, and also the silent mutation, missense mutations, nonsense mutation, only changes in one single nucleotide, they cause these kind of the variations. They have been classified generally in the synonymous changes or non-synonymous. In the change in amino acid, they are called non-synonymous. They are very expensive, costly to cause the diseases. For example, here Craig Venter sequenced his own genome 2007 in his private company, and he has lesser genome sizes about 2.8 billion base pairs, and he has about 3 million SNPs. Out of them, there are about 14,000 and that are the protein coding regions. They are present. 20% of the SNPs are never seen in any other genome. Has a unique genome sequences, and his genome sequences analyze that he does has a abnormal genes and stacking. He has taken cholesterol, high level the cholesterol accumulating gene, and he has started taking 2007 itself the stacking to thinning of the cholesterol as genomic medicine as he himself introduced, and he is happy with that also with his variation, whatever is identified. I hope every one of you are aware of this, the sickle cell anemia, because of single nucleotide change, and that has led to glutamic acid to valley at the 6 positions here. And look at the phenotype of the blood cells there. Oval shape to become sickle shape where vaccine was not able to carry. This is because of single nucleotide chain amino acid chain. Similarly, there are the microsatellite, example microsatellite, the trinucleotide repeats are there, even they have been classified depending on the Fragile X syndrome, wherein the gene is present on X chromosome, and the CGG repeats. If it is more than 54, then it's a Fragile X. Generally, up to 8 to 54, the normal individual has that. Similarly, Huntington's chorea, if it above 35, the CAG repeats, then it's end up with Huntington's chorea, and they are very well classified. The trinucleotide repeats, even some of them are present at the 5' end of the coding sequence, some of them are present in the coding sequence, some of them even present at the 3' end of the coding sequence. All these are contributing to cause the diseases. For an example, like usually this kind of example used to be used for genetic code, and here insertions or deletion, if the 3 nucleotides deleted and thereby in frame mutations, if there any of the for frame shift mutations, if one nucleotide is changed or substituted, added or deleted, that becomes a frame shift mutation. The whole coding property of the gene is affected, even though the one single mutation is happening at the N-terminal region, the whole protein will not going to be formed, and that will be drastic changes to cause diseases. Copy number variations. Generally, we do have, because we are a diploid organism, we do have 2 copies, and Because of the regional homologous repeats, segmental duplications, and chromosomal rearrangement, duplicative transposition, or non-allelic recombinations, the two copies, supposed to be two copies, will be less, one copy or more, no copy, or three copy or four copy number have been changed in the organism. This also amounts to diseases. There are several algorithms that have been used to analyze the genomic variation. Some of the copy number variation, where we did a lot of work on copy number variations studies in about 2000 genome. So these are the softwares we used in our lab, and we got many of the interesting results, and which have been already published. So, how much we are identical among us? 99.6% we are identical. Not only the Indians here, all ethnic groups, we have 99.6% identical gene nucleotide sequences. We are just different 0.4%. Some databases say 0.6 or 0.4. At least take it from 0.4 differences, which amounts to 4 million base pairs minimum between two individual differences. And this could be in a neutral position or the missense position, wherever this kind of the mutations or this kind of changes are there, that depends on, and also the kind of the genetic material we have, homozygous, heterozygous, where exactly they are present, the variation that causes the complex diseases. So after reviewing briefly about the genomic variation which causes the diseases, let's briefly go through that because the 0.4, that is 4 million base of one individual. Now we have millions of human genome sequences, exome sequences available. How to compile them, how to make the message out of that, the genome data analysis was helpful, and where our bioinformatics tools are getting there, and these tools are certainly and new algorithms are helpful for the data analysis. To understand the genetic diseases, to understand the variations, that people have started with the allelic analysis or sick pair analysis using the markers, microsurface marker, SMP markers. Later, the SMP markers have been used for the GWAS studies. There is a genome-wide associate studies. Several thousands of individuals having like a case control studies that people have studied them, meaning out of it many the positive reasons have been identified, the symptoms have been marked for the causing the diseases, and now we have the NGS, next generation sequencing, gel-less DNA sequencing, and that has also enormous data is available. All the genome exomes have been sequenced, that is available there. And exomes, the whole genome and exome data, 25,000 gene in exome data, or the whole genome data, we'll have around 3 billion base pairs per haploid genome. And there are data now specific gene panels, the gene tests are there. BRCA1 or BRCA2, like the test, their data will be available with the family members, different the individual, the cases and control studies, they are available. Even the specific gene panels are available, where the few hundred genes have been labeled, and they have been used as a panel for screening for the complex diseases. And also we have seen pooling the data. Now it's in the database available about the millions of the SLP data, polymorphism data available, about, say, told you the 25,000 transcript data, that is the gene data, even protein array, about 1 lakh protein array of specific protein products are available, even metabolomes are also about 5,000 are available. All this has to be integrated, pulled together to make meaning for a particular disease. So there are several sequencing technologies are available. They start earlier Sanger sequencing, Maxwell-Gilbert sequencing. Now they have been modified with the Geller sequencing, ion torrent, AB apply system, even complete genomics, biosciences, helicos, Illumina, and primitives, and 454 the sequencing technology. There are several sequencing the technologies available, different chemistry they are being used, and that data is available for the bioinformatics, the technicians, or the skilled person to be analyzed. And they are now the huge data is available. How to store it, how to retrieve it. Now they have to overcome this, the cloud computing services available, Amazon Web Services available, Genome Plus is available to store and retrieve this data. Now we need a person how to analyze this data. We need a skilled data analyst, data scientist, a data geneticist, a data bioinformatics, whatever you call. We don't have a skilled person for the analysis. They're looking for the skilled person for this kind of data analysis. That is one of the reason for us to start our diploma courses or even this workshop to make the at least to develop the skill and interest and to motivate them to get into this kind of field. The third level of my talk is to later I'm going to integrate that what is the current medical practices we have. Like the blockbuster concept that somebody has a fever, give same kind of tablet, somebody has the trial and error method we are following even today. Most of the diseases we have treated, we have the problems have been treated with this trial and error. The same symptoms, same diagnosis, same treatment, same dose of the medicine is being given, but there is not, their action is non-specific, non-selective, and they are not uniform. That's why many of the diseases are not able to cure. And this is the whole paradigm. What they do, they are diagnosed with the same kind of techniques. They will give some medicine, some drug. Is the drug the one course? There are 3 days or 5 days or 7 days. If they don't work, then they'll ask to take another drug. Even that also not working, then the system to the different category of drugs. So all our majority of our diseases are with the trial and methods now. And many of the drugs are available. Morning, even logo has told billions of drugs are available there. We do not know that many of them are toxic, so many side effects, and even some of the drugs are already available. If you look into that, none of the drug is 100% accurate for particular disease or particular kind of drug. One. So their efficiency is less, more of the side effects of it. About 7% of the patients in hospital experience serious drug reactions that causes many new kind of diseases. If you give the drug more, then they are having adverse effective. If you give the drug dose less, they are ineffective. We need to have a therapeutic window, and there's a lot of even drug is having a lot of side effects, but we will take the drug in the therapeutic window. That's why they give 3 days or 7 days or 5 days, any kind of antibodies for the COVID or whatever kind of infection we have. And this also drug metabolism is affecting that, and because we give the same drug to all the individual has some kind of the symptoms of the phenotype. Same symptoms, same findings, same disease, same drug. And these individuals do have differences in the single unit polymorphism, the different regions, different chromosomes, the SNP bins are there, and or there could be a non-compliance or drug interactions will be there, or none of these factors are known, but simply we're giving the drug and ended up with toxicity or more of side effects in the present day treatment. However, we do have healthcare technologies with digital medical records are available, serum biomarkers, several serum biomarkers. Now, people, if you want to go for a lipid profile, they, whether you like it or not, all the lipid profile they will give, including all the minor details. There's such kind of screen is available. And some of the identified simple single gene disorders, the defects also can be identified. And the imaging technology available, CT scans, all those images, MRI scanning is available. Even MRI expression level also it can be available. There are a lot of progress in the biomarkers for the disease analysis. And here some of the pseudo companies are available now. They say we do the genetic screening or genetic testing or genome testing, and finally I will cure the diseases. I don't think they can do that. There are many false companies are available. They just do the only chromosome analysis and they give the report that no, this is a genetic difference. Such kind of things has to be, one has to be careful to go for before going for such kind of tests. So now the focus on the clinical needs, that the bedside to the bench to the bedside, you take the samples, analyze the bench and go to the bed sample, and with this kind of information, doctor cannot act. Why? He has his own medical textbooks, they study, and those medicine only they will give, and they may not understand what is SMP, what is CNV, or how these kind of the enzymes or the proteins are causing that. But however, they do act. That's why the treating the diseases are not successful completely. So from that, I'll go to the fourth level is that what I wanted to tell for the audience now. Genomic medicine. What is genomic medicine? It is the use of information of clinical information of the patients, the subjects and the cases, and the data from the patient's genotype, that whatever the kind of analysis they are, exome sequencing, specific panel sequencing, or whole genome sequencing, all the genomic data, putting the phenotype and genotype data together and establish the strategy, stratify the disease. What is the kind of disease? Is it dominantly inherited, or the recessive inherited, or autosomal, or or the sex-linked, or multifactorial, or how many factors are involved based on the clinical data, pedigree, phenotype data, as well as the genotype data, and select the medications, available medications, and provide the therapy and initiate the preventive measures to the whole family. And this is suited for a patient at the time of administration. So that family only will get this kind of medicine. It cannot be given to all. A right medicine to the right patient, to the right dose, and the right time. That is the genomic medicine. This medicine will make an impact in the field of oncology, where the cancer causing genes have been identified. Pharmacology, what drug has to be given, rare or undiagnosed diseases, as well as infectious diseases, the genomic medicine can be given. So genomic medicine can be given any time in the life cycle. If they are diagnosed early prenatally, pre-implantation or prenatally, or in adolescence or in the adult age or in old age, the drugs can be given, the defect can be identified, it can be given at the different times. The genomic medicines not only for the old age people or the people who goes to the doctor there, this can be given if the genome sequence or exome sequence properly analyzed with the proper tools. There are already well established the multiplex tests for genome medicine. In the oncotype, there are, for example, about 21 genes which are called the tumors, and they determine the recurrence score. They use a qPCR or mRNA expression in this tumor samples, and mammoprint for breast cancer. 70 genes, the panel is available. The allo map, even for the heart transplant patients, they are going to use. There are many of such kind of the panel, gene panels already available in some of the companies, and they screen for the family who have this kind of problem. So genomic medicine is the observations, do all kinds of tests, and make a proper specific action and make the predictable response. This is not the breaking the cycle of trial and error medicine. So with this, the genomics can be equivalent to like field of pathology or biochemistry. Without understanding the what kind of cells have been, astrasia, what the cells are gone in the pathological section or in the radiations, the images, and similar like biochemical test. Similarly, the genomic sequences also can be used for treat this. So, for precision medicine, the diagnose, the focus, the prevention and treatment, this genomic medicine is helpful. What can we learn from the personal medicines? Disease risk for common diseases, disease carrier status, familial traits, or drug susceptibility, ancestry, the mutations in all the 24,000 genes, or what kind of CMUs are seen there, or microcircular polymorphism, or epigenetic factor also can be understood with the genomic medicine. So here I wanted to tell in very briefly about what work we have done in 2013 and 14 also. We found an advertisement in Times of India 2012 June, wherein a patient with having cardiovascular diseases has been online subjected for in the Apollo Hospital, Ahmedabad, subjected for multiple blockages, and for to put a stent, they take the help of the doctors there. This was the doctor, the Shamid Dani, they collaborated with him. He gave a sample of his patients. Now let me tell about the patient. The treating of the patient here, this is the profile here. He has 34 years person having all this kind of atherosclerosis, and when the parents are normal, here the two kids are normal, they did not have any kind of the problems of this, no family history, no cardiac problems in the close relatives, no consanguineous marriages, no poor lifestyles, but he had myocardial infarctions, heart attack, ischemic attack, that is mild stroke, and also severe type 2 diabetes. With one person without having any family history had that, and he had 8 blockages in the major arteries in his body, that cardiac arteries, shoulder artery, carotid artery, renal artery, and the multiple narrowing of the blood vessels because of atherosclerosis, where the cholesterol accumulates, and within 6 months, the blockage of arteries is seen there. This is kind of the image of the persons where the doctor said provided us before they apply this stunt for this purpose. There are 90%, 80% or 60% blockages there. So based on their clinical observation, these doctors, cardiologists, they gave the medicine. These are aspirin, these are the standard medicine. Generally, they give without the genomic analysis, and this is the dose they given, and this just thinking to prevent all these diseases. Then our collaboration, we got the samples, we did a whole genome sequencing and transcriptal sequencing of these individuals. And to make the story short, there are thousands of variations we could say, but we shortlisted them. 14 mutations have been very severe mutation identified, 11 of them non-synonymous, and 3 of them are insertion. All these were found in exomic regions. And these are the genes, all the details are there. You need not worry about this are the genes now. Based on this, our genomic information, we send our genomic information, they re-describe, attack the receptors, the processing of the one, even this lamb was one of the gene was defective, and that was the one controlling the hundreds of genes, the down-regulating genes, and they could re-describe the drug, and the patients, the blockage of reduced to some extent, they compared to the world. This is one of the work what we did in university. The major people involved in this, two of my students, Dr. Avinash and Dr. Dinesh. Dinesh got the money from the NITE, and we could able to do this work in our lab. Still, the data has not been published. Okay, now the last part where I wanted to summarize with what is the current practice of genomic medicine? Is already there, and we have to go for it, and what is the status of that, and what are the new issues and challenges for the genomic medicine. So who are interested for the I'm not I'm taking the genomic medicine for the person who do not have the money, and who are interested, who have the problem, we wanted to know proactive approach for the healthcare eligibility. Elite athletes, they can predict their sequences, and concern with the hereditary diseases based on family history, prenatal testing, and concern for the environmental exposure, some of the exposed environment, what kind of mutations are there with gene, even diet and exercise customization. Now people are going for nucleocode genomics, and there also they go for genome sequencing, microbiome, or epigenome sequences. The people who are interested in this, they are already into the genomic medicine. 2014, Obama agreed in the era of personal genomics that is out of 41 drugs which were available in the market, 8 were targeted to the genetic mutation, the data, and which they have been tested for efficacy tests or pharmacokinetics, and they have been approved from the FDA of America. That was the beginning of the genomic medicine. People have started taking on this, and this is increased. Genomic medicine has increased in order of magnitude. Now people are taking on this. Look at I hope most of you are aware of this. The Angelina Jolie, her mother and aunt had the ovarian and breast cancer, where the BRCA mutation have been seen. She also tested the DNA, and she also found the positive for the mutations. Then she went for the surgery, double mastectomy, and she started taking the treatment for this monoclonal antibody for the BRCA, as well as the HER2 gene, which is a 25% of the cancer patient had the HER2 positive, and the molecular antibody for HER2 as well as BRCA to taken, and she is in complete treatment. She has not seen any of the so far no reports there that this has been treated with this. The herceptin is a human epidermal growth factor, the HER2, where I told you about 20% of all cancer has these mutations, and they will go for this has become a regular treatment for any cancer patient. They give it is the monoclonal antibody. This has become a routine part of the clinical diagnosis of breast cancer, and this is given for many patients with HER2 positive. And another interesting case where the genomic medicines are given is the familial hypercholesterolemia. It's autosomal dominant diseases, where the atherosclerotic diseases, blockage of the arteries because of accumulation of more of the cholesterol. The frequency is 1 in 200 to 500, and they identified in the patient the 3 mutations: LDR receptor, FOB receptor, and PCSK9, the enzyme has detected. Based on the studies in the Daesh population subject, about 490, 13 and 1,537 case control studies, they gave for 10 years or 15 years the statin, the drug for thinning of the cholesterol. Look at after treating with the statin, with a simple statin drug every day, the improvement of the cholesterol accumulation is increased even to 80%. The success story. The one who has not taken here, they even 20% of that the success. That means they are not able to survive. So a genomic medicine, if they properly identify what is the defect, certainly is useful. Recently, that is 2012, the UK Prime Minister, after looking into the increasing of cancer patients in UK, the people with National Health Services, the UK is one of the best health services in the whole world. Next to that is the Canada. And what they did was most of the patients are, lakhs of patients are with the cancer patients, and he ordered the government, or he take the money from the government, created the England Genomics Company as a government company, and 2012 they sequenced the 1 lakh cancer patients of the National Health Service individuals there, and 2018 they completed, and this is our 1 lakh genomes, 70,000 the patients and the family members, and data 21 petabytes data is available, and there have been different 1,500 staffs worked on that. The researchers, scientists, 2,500 scientists for this 6 years they worked. They completed the whole genome sequencing, and the 13 centers they established to treat these patients, cancer patients. 13 centers in the whole UK. This is one of the success story. I think UK is going to be a world leader in genomic medicine. And these are the cancers they identified. They identify the mutation, the major mutations they identified, the target of the mutations. The treatment have been started in 2018 itself. And other genomic medicine for the diabetes, the single gene detect, that is MODY 1, 2, 3, and for the Parkinson's, then the genes genomic medicines are already available. So now genomic medicine in practice for prevention, for efficacy, and the safety. Different drugs have been identified, they are being in use. Last year, the problems of the kid, Cora, son, she had a problem of bad eyesight and struggling in schools. She was 5 years old, she had a problem. They did test 1,500 disease gene in the panel. They are not able to find. Finally, they found the one biotinase enzyme is defective, which degrade or which convert the beta B vitamins there. So now she is being under treatment of daily biotin tablets is being given. She is thriving better than the earlier one, and now that the even 1,500 screening did not find only one. This biotinase a single gene defect they identified in the skin. So now the new paradigm for treatment with the genomic medicine is that screening, predisposition, what is the problem in the family and the population and the individual, wherever is possible, then monitoring them and by preventive measures and whatever the targeted treatment can be given, this is possible now. There are a lot of potential benefit now. Instead of reactions or side effects, is a more of preventions. The medicine is customized for the individual and they are more effective and avoid the side effects of these patients. Now with the genomic medicine, people immediately say it's very expensive, they costly, and also yes, to some extent it's costly. And now a lot of changes has to be done. Now, as Raghu has said in the pharma, the technology, a lot of changes are happening. Here also a lot of changes has to happen to the successful of the genomic medicine, like the payer, the expenditure is increasing now. Physicians has to be trained. They are not understanding what is genetic. Many times when the patient comes there, oh, this is hereditary. They just leave it there. They will not go into detail aspects about that, and whatever they have medicine they give, and with all the doctors. It's a very, very sad story that our medical school in India, they're not being teaching the basic science of genetics there. Very few. They even one chapter, one lesson, they are there, they will not be there. Even I have a lot of interactions last 30 years as their doctor, they say, sir, it's not very critical, they will not study. That is the pathetic situation. Except few AIMS and even St. John's, they do have some kind of concern with this. Maybe some other medical schools. Patients, certain drugs are expensive. They have to go for it if they want to treat the disease. Authority, how to deal the complex situation, still we have to know, because this our organization, government organization, health sector, they have to understand this. And diagnostic labs, they have to do the more of bed financing. Industry, very few. We don't need millions of drugs. Why do we need them? Very few. We have about so far they identified 7,000 gene mutations in human. Maybe about 10,000 or 20,000 drugs is sufficient to treat almost all the diseases. We don't need so many drugs. We need not waste more money for the drug industry. Even computational methods can be used, and first one has to diagnose properly. The major problem, understanding, is not there. Problems of diagnosis is there. They even many symptoms, they immediately predict, the doctors or whatever, the expert they predict, because many symptoms are shady there. The clinical diagnosis, the phenotypic diagnosis is very essential. If those things have been done properly, almost all the diseases can be treated, even if you can go to the extent of curing the diseases. What are the challenges? We have to change in our culture, like practitioner populations, we have to go for such kind of tests. Require change in medical data analysis. The huge data will be available. One should know, one should understand the patterns. What kind of pattern? Oh, if this is the kind of situation, what analysis one has to do, what kind of medicine we have to think about that, and also the required change in the diagnostic approach. We need not test all the machines in the hospital there. Whatever is needed, we have to test. So that's we can reduce the burden. Diagnosis, proper diagnosis, very needed. We have to make these changes. So genomic medicine is a predictive, preventive, personalized, and participatory rather than reactive. This is the outcome of the genomic medicine. The role of physician has to change. Even the patient has to be educated, has to give that what is the factor. Many times when you ask the doctor, they say, you take the drug whatever given. I'm not getting a toxic. Even now people say that attitude has to be changed. They have to explain, otherwise they will bring the tens of pages that to sign in a critical condition, you sign it because we don't want to take care of any of this, responsibility there. So they have to explain what is the problem. That is lacking in our medical system, medical school. They have to drastically change. Broadening the definition of patients simply we can, so we have to broaden and understand what exactly the patient has a problem with. And now the social media and e-health will influence the healthcare. So when you do all these things, ethics is very important. Without ethical care, you cannot do. You cannot directly test some people do some research papers, they publish paper in Nature Genetics or PNS, they claim that no, we'll cure the breast cancer, we'll cure the colon cancer. It's not happening. It has to be, that is a general one, some cell line, some model system, they are shown a positive. Doesn't mean that will going to cure or treat the medicine. The long way to go for it. Okay? So one has to have proper ethics. One should know how to handle the patients. They are not the guinea pigs. So we do have a lot of limitations. One has to understand, the people has to understand, clinician has to be educated and more responsible for the government and the society is there for a lot of limitations. Are you ready to take the genomic medicine? Whether we like it or not, it is already there, it is going to be there, and we have to explore that. The only fear for the genomic medicine, you might say, oh, if I already know what is going to happen to my, I may not get a job, I may not get married and so on. If you know early defect, you can manage yourself properly. Instead of hiding, pretending, we should know what is true. That way you can live. That this is the kind of thinking, knowledge, kind of the openness should be there. Otherwise, how long you can hide this? The family. So, future of genomic medicine, even now we do not know complete fundamentals of science, or our DNA, or genetics, or genomics. The complexity of the disease, risk, roadblocks, signals, and therapy we do not know. We need to know more about it. Comprehensive catalog of all the molecular changes, lesions, or the diseases that we should know. Understanding the genetic basis of nearly all Mendelian disorders, rare or the common, and then diagnosis and cure. One has to understand that. Finally, to expand in basic science. Foundational resources and technology for genomic medicine is needed. We need to have a basic science. Most of the time, we will forget about the basic science. That is the strength. Now the course is giving here the foundation, and if you get motivated, you can go for any other advanced technology and so on. So I am closing this with the take-home message of any faculties are here, think about revising syllabus. Genetics should be taught, practiced, propagated as a foundation for all life sciences. You can introduce, now already, I think I was telling to other person, the BSc genetics pass students have been recruited for high schools. Government has agreed, whole throughout the country they have agreed. Now this has to be taken very serious to make them to understand. Genomics should be treated as one of the specialized fields, especially in medical schools like pathology, oncology, or biochemistry. Neither they teach digital genetics, they hardly understand the genomics now. But everybody use the terms here and there, they copy from the internet, they present it here, they say, yes, we know. And after talking half an hour, 1 hour of my medical school doctors, Ramchandra, what you said is gone above my head, I did not understand. After talking half an hour, 1 hour to them. This is the situation of senior doctors I face now. And finally, your take-home message is genetic diseases are treatable. Look at the Juliana there. Many of them, the Cora and the kid there. And manageable and preventable if diagnosed properly, preventable. Major problem is the diagnosis and understanding as a subject. We don't understand the fundamental. We have a semester system and 4 months course. We have only 2 months working. After 2 months, even 16 hours portions, we teach only 8 hours. Out of 8 hours, many of them are missing and not properly. We'll give questions, even 35% or 40% they write examination, they graded them passed. Where is they? Do they understand the subject? Are we really doing the right kind of thing? Our society, I'm talking about, not talking about the teachers, our system. Everybody blame on the system. Who is system? Who is creating the system? Our NEP syllabus, who is making that? And many of the courses are not being properly taught. Now already we announced that we have first to introduce NEP in Karnataka government. This is my group, Avinash, and look at here, Malini is also there. She was the one first struggled hard in the department to start human genetic research from her pocket money, from her salary in 2000. Now we have more than 20 students have done PhD in human genetics. Her work was that about just pedigree analysis of Down syndrome. We have contributed, even people upheld that our grandmother age is responsible to cause Down syndrome in Indian families. Young woman in India getting Down syndrome because of her grandmother age is well established. We made a publication, medical genetics, BMC medical genetics. Even people quote that we have highest citation for this also. That is the starting one. Now, even the latest one where the microbiome data we analyze in brain research, and that's also been started quoting about it. I mean, I'm not telling only these two members, all have contributed and they selected me, and I could able to help them to do the work. So, along with my PG diploma students, they are very humble. They stayed with me, they worked with me, they discussed with me, and also many of the project students, MSc project students, they got a good publications by taking this kind of problems, even MSc, other students also. I profusely thank all my colleagues, my students. I forgot here my collaborators, doctors, and my university, which has given place for me to do some kind of thing. When they are not allowed to do the DNA isolation, we have reached this stage. Don't you think that this is an achievement here? With this university where you struggle, even you struggle hard to get good water, good power supply, good drinking water, good power supply, good facility there. We do have some facility, but there, but still we could able to do some justice in science. Thank you, all of you. Please, you can interact now.
Any questions from the students? One of the questions.
Any kind of presentation is more time for the discussion should be there.
No questions, it is lunch hour mostly.
Always my problem is that.
Yeah. Anyway,
I will be around. You can ask me, and also if you look at my first slide or I have my email and my website. You can look into that. The features and you can interact.
yeah. Sir, I think it is the concluding session of the morning session. Thank you very much for a nice lecture. See, we are actually in the era of the professional doctors. Right? And now we have introduced probably the terminology genomic medicine. So we have to look into the genomic medicine now, and they have simplified your probably presentation in such a way that commencing from the basic genetics. Yeah, you are a basic genetics man, I remember. Drosophila genetics, then human genetics. Now genomic medicine. Yeah, you are actually you talk more about the evolution, but you are academically evolved so much that commencing from Drosophila genetics down to genomic medicine. Wonderful. Yeah, very nice lecture. I think the students community, what I just request is that, especially the younger generation, they don't know him because we have joined the department very recently. So, especially with regard to the SPP concluding session, I just request that he will be available in the department from morning 10 o'clock, 10:30 to afternoon. You can just definitely keep him and discuss with him any kind of clarifications with regard to the not only with regard to this genomic medicine related to the academic matters with regard to the genetics. So please interact with him, sir. I know I have already introduced you in the morning. You have done a wonderful job from the beginning. 1980 as a student you have joined, and now after 42 years of interaction with you, I have seen how much you have involved in the university, how much you have struggled to just establish the department, and definitely thanks to you, a nice lecture you have delivered today. So give him a wonderful so I just want to see that Dr. Amrutvalli Madam has to just deliver the lecture now, but I think it is probably she will take care of that in the afternoon session. So thank you very much once again. Thank you.
I need to look into this thing to carry further. So wherever the curriculum are there, you have to consider this kind.
Okay, good afternoon, everyone. Now there is a couple of announcements. MSc first year and second year students, there is a strike and they came twice and they made us to close the department. So you people can disperse from here. Afternoon, we don't have a practicals today. Okay. And participants, you have to come down to the department. And we do one thing, you faculties and diploma students will take you, and you have to start from, you go from a BIMS gate, that's it. Because still few strike people are there, they are not allowing anybody to move. So you take them from this gate and Hunsu Road, then go to BIMS gate and then go to the department. Only participants and diploma students, rest of them please disperse. Otherwise, if you come to the department, they'll make a big hangama. Okay, thank you everyone for the patient hearing. It's a wonderful talk. Because of too many things, I was unable to concentrate completely. And now I will disperse for this one. Next session will be done in the department, and all 7 days will continue in the department. Thank you.